by this platform we can use.
Start live streaming. Yes, sir.
Good morning, sir.
Yes, good morning.
Sir, you can share the video and audio from your end also.
Okay.
Or if there is any difficulty for technical reason, then you can send to us. Then we can play.
Okay. From share option, if I'll go, okay, just show me. We optimize. Actually, there is one option in the MS Word to share the audio as well while sharing the slide. Yes. Okay. Now it can be done.
So we are going to start by 2 minutes.
Sure. No issues. Okay.
A very warm good morning to Dr. Amit Alexander, sir, and my dear participants. Today, we have with us Dr. Amit Alexander, who is the Associate Professor of Department of Pharmaceutics, National Institute of Pharmaceutical Education and Research, CIPET, Guwahati. He has more than 12 years of teaching and research experience. His research experience is in the development of chitosan-based injectable hydrogel, thermoresponsive hydrogel for breast cancer therapy. He has expertise on pathophysiology of Alzheimer's disease, nose-to-brain drug delivery, and application for in situ gel for nasal delivery of anti-AD drugs. He has expertise in vitro and in vivo thermokinetics estimation of nanocarrier system. He has expertise in practical skills in spectrophotometric like UV visible FTIR analysis, HPLC, nanocarrier systems, in vitro release, and permeation study. He is a design expert in statistical analysis using ANOVA response surface methodology. Sir's research interests are mainly on Alzheimer therapy, nose to brain delivery, nanoparticle incytogelations, injectable hydrogel, breast cancer, and he has a highly significant research publication with citation index 2839, having H index 28 and a significant item index of 54. Some of his association is his association with different organizations, vast different professional bodies, but I am just going to mention few of them. He is a life member of Indian Pharmaceutical Association. He is a life member of Association of Pharmacy Teachers of India. So thank you very much, sir. Welcome to this session, and sir will deliver a lecture on polymeric materials in pharmaceuticals. Over to you, sir.
Okay, so thank you so much for your introduction and good morning to one and all. Um, I will quickly share my slide. Please let me know if it is visible to you.
Visible, sir.
Visible, right? Okay.
Yes, sir.
Just a minute. So I unnecessarily how to get rid of these panels? Like, can I remove this video?
Sir, here a slide is coming.
Okay.
We are viewing your slides.
Okay. Because I am viewing this interface of WebEx. So
Yes.
that is the thing. Okay. So I hope now this is visible to you. And
Yes, sir.
yeah, once again, very good morning to all of you. Basically, as far as this theme of this faculty development program is concerned, I hope all the faculties are there in the program, right? Am I audible?
Yes, sir. Almost
Yeah.
we have 100, more than 100.
Oh,
Now 55 is there. So they will
excellent.
join by, sir, 2-3 minutes.
Okay, okay.
You
So
can
can I quickly know the area where all the faculties are working, like apart from teaching, if they are
Yes,
doing
it
research?
is an interdisciplinary type of nanotechnology, chemistry, physics, pharmaceutical,
Okay.
biotechnology, mechanical. It's mixing.
Excellent, excellent. So
Yes, sir.
before going into the depth of the presentation, it is wise to know the audience with which we are dealing with, because accordingly we can, deliver the content actually. So basically, this particular theme which was given to me was of a very interesting topic, and to me also, because this topic tells you about the polymer matrix based nanostructure. So this matrix based nanostructure for the targeted drug delivery application. But yes, I have chosen this particular topic for to map with the theme, that is the polymeric materials in the pharmaceuticals, because this is what more important to know about your polymeric materials and their role of, you know how best we can tune those polymeric material, and we can use it for the drug delivery. And basically in our institute, what we are doing, we are in a process to establish a good delivery system, so that for any kind of ailment or diseases, the particular drug which is responsible to cure that particular disorder can be delivered to the site of action. First of all, its bioavailability should not be hampered, and also at that certain stage you need to think about the cost also, because whenever you are into some kind of advanced, you are deploying some advanced technology to your delivery system, it's very important to also monitor the cost, so that it can be transferred to the industries. Technology can be transferred and it can be very well uptake by the the particular audience and mankind, because if your cost of the medicine is too high, it will not reach to the particular disease patient, and uh, that visibility of your work will be not up to that mark. So while formulation, we have to choose certain kind of polymers which are basically should have a very good kind of property which you have decided to make, and at the same time its cost also should not be at too high, because when we are shifting from the conventional to the nanotechnology and from nanostructure to targeted, that means again you are deploying so many technologies, and that's why all the technology developed under the criteria of targeted and nanostructured are very far away from the common people, but yet they are life-saving formulations in some cases where conventional therapy will not work. Okay, so continuing with this polymeric material in the pharmaceuticals, I will go ahead, and this is the release mechanism we are studying, that is the control release system. So basically, what is the meaning of control release system? So these particular control release systems are the systems which can be tuned, or which can be controlled by the formulator once it will be ingested inside the body, because as you know, the basic example of tablet and capsule. So tablet and capsule, generally when the tablet is taken up by any person or any patient, so what happen is, after 1.5 hours, the tablet will go inside the stomach, and it will get disintegrate and dissolve, and after that the absorption will occur. So by this time it will take around 15 to 30 minutes for the whole process, and after taking tablet, after 15 to 30 minutes only, the patient will get some relief. Why? Because there is a process which is the disintegration and the dissolution which need to happen, and that is why there is a time lag. So to remove this time lag, what is an alternative? So alternative is injectables, because the injectables or the IV route, the intravenous injections. So what they will do, they will directly deliver the drug in the systemic circulation. So systemic circulation, here I am telling about the blood circulation. So this is the one technique by which we are going to make uh, our drug available into the systemic circulation. So since it is a different kind of uh, science background and engineering background blend is there in the audience. So just for your basic information, I would like to tell that for any drug, the action can only be achieved when it is available in the blood. So I am taking a just normal, not the too much technical terms. So this blood, we are calling it as the systemic pool. Is it visible, whatever I am writing? Is it visible to you?
It is visible, sir.
Okay, okay, fine. So drug can only be able to give the action when it will be going to the systemic pool, because this systemic pool will take up your drug and it will deliver it to the particular site of action. So this is the basic theme of drug delivery. Okay, so since our title was targeted drug delivery, and I have chosen the kind of polymers we are dealing with. So this is the part of main objective of any formulator, any pharmaceutical formulator, to deliver the drug to its site of action so that action can start immediately. Okay, so if you take the example, afterwards we can discuss also this one. So there are a different class of generation to generation this particular drug delivery systems, and the control drug delivery systems particularly have been developed, because earlier when the first generation medicines we are considering, so there was an advancement in the modification of the conventional dosage form, because this conventional dosage form, take an example of the tablet only. they're supposed to deliver the drug up to 1 hour only. Why? Because it has to go into the stomach, then it will break down, it will form a solution, and this stomach is surrounded by so many blood vessels. Okay, and the duty of these blood vessels is to keep the temperature of the stomach up to 37 degrees Celsius. So that is the general phenomena. So when this drug is disintegrating and it comes into dissolution state, then it will go to the systemic circulation from this membrane. Okay, so this is the bioavailability. So for this, it has to be get into this step after breakdown into dissolution step within 1 hour, and this tablet should be able to release the drug into this period of time. So this is the drug release mechanism. It will first break down, disintegrate, and then dissolve. So this is the general criteria for any tablet or any formulation, so that it can be well taken up by the systemic pool and it should be absorbed here. This is what our absorption is. So this is the conventional dosage form. Now, after this, there are certain dosage form which need an immediate action. So for that, this particular process, disintegration, because it takes almost 20 minutes time. Okay, for a normal thing, because the tablet will absorb the water and after that it will swell and it will disintegrate. Okay, it will break down into different particles. But if you are using certain polymers like super disintegrant agent, immediately, have you seen tablet Disprin? This is a very common tablet we all have seen. Uh, anybody of you have seen the tablet Disprin? Hello?
Yes, sir.
Yeah, so the property of Disprin you can see, because if you put it into a glass, it's supposed to disintegrate first of all, and that tablet has been made. There is a reason to make the tablet into this form, because this tablet always the dissolution will occur after this step. This is the first step, disintegration, and second is the dissolution. So once your drug is in the solution form, it will immediately get absorbed through the systemic circulation. Okay, so now the concept behind preparing this super disintegrant or immediate release tablets are that your tablet should be able to disintegrate and convert into solution into glass itself. Okay, so why we are not preparing the solution, because the stability is a major concern. Okay, if it will be there in the solution form, you cannot keep that particular solution up to longer period of time. Okay, because some reactions will happen, hydrolysis will happen, something deterioration is there because of the presence of water. Now, to avoid that, formulator had designed a tablet wherein you can immediately before consuming you can convert it into solution form. Okay, so and this solution you are taking up quickly, so it will be in the solution form only. So this will give a quick absorption. So generation to generation like this, the research has been done, and we want some drug like immediate release. We want a drug that should release the drug up to 8 hours. Then another technology, we want to reduce the time of dosage form. Like if the doctor prescribes you some medicine, he suggested you to take this tablet 4 times a day. That means 4 times means you will forgot, you might skip one of the dosage regimen. So this will not give you proper appearance of the drug in the systemic pool, because 8 hours is a general one. We have increased the drug from 1 hour release to 8 hours, from 8 hours to 24 hours to 72 hours, and there are certain delivery systems which can deliver the drug up to 3 years. Okay, we will discuss it in my next stages. So likewise, this generation to generation, this modification has been come up, and initially these drug molecules are very small in size, micro level or nano level. We are converting it this delivery system. but yet now the present generation is working on the delivery of macromolecules like protein and peptide. Okay, so that it can be very well taken up, and also we are in a process to target certain site like blood-brain barrier. Okay, so this blood-brain barrier need to be targeted because this is the barrier which will prevent any substance to come inside the blood to the brain region, and it is separating the brain region from the entire systemic circulation. So these are the general introduction about the drug delivery system. Now, considering these conditions, we are formulating and we are developing certain kind of delivery system. So this evaluation of certain controlled drug delivery systems was, started in the year of 1950, and since 1950 uh, these particular modification have been taken up, and you can see the basic of the controlled drug delivery system. This is I'm don't get confused with the conventional one, because conventional after that, these conventional were there before 1950 also. Okay, but after that, if we want a sustain or the controlled drug delivery system, that means if we want a release of drug over a longer period of time, so what was the uh, basic improvement? So you can see 1950 onwards the research have been done for the oral delivery of, only they have reduced the time to twice or once a day formulation. If there was a need for 4 times a day a drug need to be taken, they converted during this year from, they have reduced the dose frequency from 4 times to twice or once a day dosage form. Transdermal delivery system. So transdermal delivery system like once a day or once a week. Anybody knows about something about the transdermal delivery system? Anybody can, uh, if you have some idea, can you share? Okay, so this transdermal drug delivery system is generally a kind of patch, okay, which you put in the skin. It will be like a patch, and the upper layer will protect this patch from kind of environmental damage, and it will be, a patient can apply on their skin, and they can walk around, they can go anywhere, and it will release the drug based on its tuning up to certain uh, uh, time periods, more than hours. It can be for days, it can be for week, it can be for month. Okay, uh, suppose I'm giving you an example. A lion is there. Okay, so if this lion got injured in any cases, and this lion need a medication also. Okay, so we cannot give this lion a medication to save his life, like a dosage regimen of twice or thrice a day or once a day. Okay, because you cannot give him a proper dosage form, and you cannot give a certain cure frequently. Okay, so for those kind of large animals, big animal, and to particularly deliver the drug, suppose some injury is there. So what the best thing is, we can put that particular patch system into the skin of that lion, and we can make this drug available up to longer period of time, so that these kind of big animals, and I'm taking this example because this dose drug delivery systems is for veterinary also, not only for humankind. To make it more easy to understand, I'm taking an example of lion, so that you can realize it's, yes, it's very tough to treat these animals also, and these animals also require some medication. Okay, so for the lion, what the thing is, you have to tranquilize the lion, the animal, and after he get fainted, then you can apply the dressing, and you can give the dosage form. So there are certain dosage form like serosopeptidase, antibiotics, so you can give, you can put in, load into the patch, and those delivery system can be delivered to the particular subject over a longer period of time, and this is the advancement. Okay, then Basically, the study has been initiated on the drug release mechanism. What are the release mechanisms? So basically, there's a dissolution and there is a diffusion release mechanism, osmosis and ion exchange. So these are the release mechanism which will tune up your uh, drug release profile. If you go to the second generation example, so second generation example is the in situ gelation systems. So these are some kind of polymeric delivery systems which can be triggered by external stimuli. So there are certain, and that's why their name is smart delivery. Okay, these are the smart delivery system. They will give the release from the dosage form in a zero order release system, and also a combination of zero and first order release. Okay, there are certain smart polymers and hydrogels which are environmental and sensitive self-regulated release systems. Okay, and they can also deliver the peptide and protein delivery as well as the biodegradable depot. In the same era, there was a vast development in the field of nanotechnology also. So nanoparticles have came into picture, and it can be very well tuned up, and it can be binded up with a ligand to target a particular organ. So these nanoparticles deliver the drug into the tumor site, and there they can also be exploited for the delivery of gene also. Okay, so now the basic the research which is going on, and still it is on the uh, platform, that is the modulated delivery system, which consists of on-off insulin release, glucose sensitive release, and targeted delivery, particularly for the anti-cancer and siRNA. So these targeted delivery system, I will tell you one small example of this targeted delivery system so that it can match with the theme also. Suppose a polymeric material is there. I hope you know the orientation of the surfactant or over a critical missile concentration like CMC. If you take, so this CMC is a critical missile concentration which will form the orientation of the micelles, and over these micelles, over a synthesis process, the silica source is getting adsorbed over this particular body, and they are generating a delivery system which is having many pores. Okay, these pores are so small, into nano range of 2 nanometer to 10 nanometer, and these particular delivery systems are known as mesoporous silica nanoparticles. This we are developing in our lab. So these pores can, after this preparation of pores of the this particular nano formulation, we are loading the drug. Suppose some anti-cancer drug, paclitaxel, it has been loaded into this particular pores, and it will be, after this loading, we can cover this pores with a gatekeeper. So those gatekeeper are MnO2, okay, manganese oxide. So these can be well placed over these pores in a particular synthesis, and once the delivery system has been ready, so wherever in the body a tumor is there, we know there will be normal systemic circulation in the system. The pH will be around 7.4, but yes, wherever the tumor is developed inside the body, its pH is little bit on the lower side, so 6, okay, 5 to 6 range it will be. So this particular manganese oxide will get converted into manganese, and it will dissolve, and it will remove from the pores in a pH of 6. So whenever in the systemic pool, this drug delivery system will come in contact with this region, so what will happen, this gatekeeper will remove, and the internal drug from the pores will be released over a particular site only. Only the tumor cell only the drug will be released, not in the entire body, because this gatekeeper will not be dissolved with the pH of 7.4. It will only be dissolved in a pH of 6. So this is basic strategy I want to tell. Like this, there are so many kind of nanoparticles which have been made and developed during these recent years. Okay. So before going into this, I will like to show the transdermal patches also, how it is looking like. You can see these are the
Transdermal patches are innovative
therapeutic
drug
systems
delivery systems
where
for an easy delivery of a
to
wide
reduce the
variety of
frequency.
pharmaceutical active ingredients.
This audio is audible. This audio is audible.
It is audible, sir.
Yes, yes. So you can see this patch. So it's very convenient. It become very convenient for a patient. No need to remember also when to take the medicine and when the second dose I have to take, and because this profile will the concentration of the release of the drug will not be maintained if you skip any of the dosage form. So you can see the function of this particular patches, which are layer by layer prepared, and each layer is having their significant features by which we are controlling the release of the drug over an extended period of time.
The basic components of the transdermal.
So you can see there are three layer basic one is the baking layer, and second is the drug containing adhesive matrix, and the third one is the adhesive matrix. So this adhesive is responsible to place the patch over the skin, and this particular middle layer is responsible to where we are loading the drug also, so that it can be penetrated from the skin, and the top layer is preventing this entire patch from the external environment.
Patch are an outer backing film of plastic or foil and a pressure-sensitive adhesive that anchors the patch to the skin. The drug is dissolved or dispersed in an inert polymer matrix that provides support and the platform for drug release. When the patch is applied to the skin, the drug is released and penetrates into the skin. It is a continuous process that can last for several hours up to 7 days. As the drug moves further into the skin, it is absorbed into the local capillary system. The bloodstream then transports the drug molecules to the site of action. For the patient, the administration of drugs.
Okay, so this was the short video to make you understand what these transdermal drug delivery systems are. Now you can map it with the example which I've given to you, not with the human. Human also can be, it was the example with the human. Consider the same thing with the animals also. So it become quite convenient dosage forms in case of large animal or some dangerous animal where the medication is required to save the life of that particular animal. Okay, so over the time, these control release systems have been developed, and you can see this particular diagram where you will be able to see what kind of strategies and what kind of routes we have opted over the period of time to prepare a particular drug delivery system. So drug delivery systems is only not only about the conventional one, but it's about the micro level also, which include both macro and nano level. And also apart from this, there is a strategy has been opted to choose the smart polymers, smart materials, as well as the polymers, and these are the particular delivery materials. Drugs, you can see small hydrophobic chemicals, protein and peptide, siRNA are can be delivered through this delivery system, and the release mechanism, which is to be monitored for the control release systems, as I discussed earlier also, like diffusion, dissolution, and exchange and osmosis. And this osmosis is the thing that means it will osmogens are there, so it will withdraw, it will take up the water from the outer environment, and it will swell upon its contact, and it will squeeze the internal implants also. I will show you this thing in my later slide. So these drug delivery routes are ocular, nasal, oral, pulmonary, transdermal, and parenteral. Apart from these routes, also there are several other routes which are being used to deliver the particular delivery system. And this entire delivery systems, once it will be made, it is being evaluated on the basis of its release kinetics, whether they are giving a certain kind of kinetics or not, and so that we can designate those things as a controlled drug delivery system, because their rate of release should be predetermined. Okay, then there is the IV-IVC correlation, that is the in vitro and in vivo correlation. Whatever the results we are getting in the lab, that should be mapped with the results we are getting with the animals or with the subjects. So small animal experiments are done, and we are seeing the biodistribution and targeted delivery. By performing the experiment on the animals, we can monitor both these things to see whether this drug is, suppose if a drug is made to delivered or to target in the brain region. So as I told you, there are in the body is divided into two basic compartment. Like the if I take the central compartment, it include the blood, and now there is a membrane, blood brain barrier, which separate the entire body from the brain region. Because if everything will come to brain, it will be very difficult, and it can cause a very distress and discomfort things to the patient. So to avoid those things, all the exogenous substances from the entire region of the other part of the body is separated with this particular vital organ, that is the brain. So to see the same thing that drug has available in the brain region, what the experiments are going on the animal is, after administering the drug delivery system, the animals is been sacrificed and different parts we have to isolate. So we will isolate brain also, and we will see whether drug is able to reach in the brain or not. If drug is not at all present in the brain after few hours, that means whatever the delivery system we have prepared, that is not at all working. So there should be a concentration of the drug in the brain region. So these small experiments will designate whether your drug is able to uh, get targeted or not. Okay. And also we are observing the pharmacokinetics as well, which is ultimately giving you the entire dosage regimen to be decided for the final preparation of the dosage form, and it will be further taken up for the clinical studies and clinical application. So this is all the development process, and there are certain polymers, you can see, which are used for the control release systems. And these polymers are used for basically tuning the particular delivery systems for certain kind of element. Like you can see, the first one example is polymers which are based on the polymerization. They are divided into two categories. One is the condensation polymers and another is the addition polymer. Okay, so examples of these condensation polymers are the nylon and bakelite. Okay, and for the rest of the other polymers like polyethylenes, polypropylene, PVC, and Teflon, etc. And apart from this, if these polymers are categorized on the basis of the structure also. Okay, so based on the structure, they are linear polymers, branched polymer, and cross-linked polymer. Okay, apart from this, the polymers are categorized on the basis of their molecular force as well, like the fibers, elastomers, and the thermosettings and the thermoplastic polymers. Okay, so this pointer is visible to you, whether is this visible?
Yes.
Okay, fine. Thank you. So if you see this particular no, different class of polymer, so this is the basic polymers which we are using for controlling the release of the system. Again, for your just for example, if you take the conventional dosage form, you don't have any control. Okay, but if you are preparing a controlled drug delivery system, you are having a proper control on the release of this particular drug. Okay, so like if you are adding ferric content in your formulation, suppose nanoparticle, so this ferric content, what it will do, suppose the patient is having a injury over a particular site, so from the external force, if you keep a magnet, the drug is moving, the nanoparticle which is having the drug and the ferric content, it will move around the body. Okay, but wherever this, it will get a magnetic field. So what it will do, it will localized here, and since the entire nanoparticle, they will do, they will accumulate over the site of action. So what we are doing, we are providing a release, or we are controlling the nanoparticle movement and its release by external force. Okay, this is also just an example, and this is known as magnetic nanoparticles. Okay, This is example of magnetic nanoparticle, so that we can control from the external source. So this is also a development. And there are certain polymers which are based on its organization, like the natural polymer and the semi-synthetic polymers and the synthetic polymers. So if you see the natural origin of the polymer based on its origin, so the protein, polysaccharides, and natural rubbers all belongs to the category of natural polymers. Semi-synthetic polymers, HPMC, CMC, cellulose, and diacetate, etc., are the semi-synthetic polymers. And synthetic polymers include PVP, udrazine, and PEG. Okay, so and based on the solubility, the polymers are divided into two groups, like hydrophobic polymers and hydrophilic polymers. Okay, so these are the examples of hydrophilic and hydrophilic polymers. So basically, and others include the dendrimers. Okay, so these are the different polymers which are used for the classic in the pharmaceuticals for the preparation of control release system. So as we discussed earlier, so basic in a broad way, we can see the release behavior into form of diffusion and dissolution. So these drug release mechanisms are responsible for controlling the drug release from any kind of delivery system. So if you take the example of degradation control system, it includes two kind of erosion. Okay, because degradation means at the end of the course, the particular delivery system will get degraded. Okay, and the erosion of that particular matrix is the responsible factors are the chemical erosion and the physical erosion. Apart from this, the second is the diffusion control system, and the third category is which is controlling the release mechanism for controlled drug release systems are the solvent activated system, and another one is the biodegradable system. So into if taking the example of the degradable control systems, this is a general diagram which is showing the outer membrane. You can see here, this is the structure. Earlier the drug was embedded inside the matrix, and what happened is once it will come into contact with the fluid, or you can say, for example, I'm saying as water. So what will happen is this particular erosion will occur. Okay, so it this erosion will further degrade the polymer. Okay, this will further degrade the polymer. Okay, so and the basic erosion occurs due to the presence of the enzyme, different type of enzymes. It will break down these bonds of this polymer. So the movement of this particular water inside the polymer matrix triggered this hydrolysis process also. Okay, so this hydrolysis and the breaking of the bond by the enzyme, these are the basic two phenomena wherein we are observing in the case of degradation control regular system. So this inside cavity is the drug polymer core, and you can see this particular molecules are coming out of from the matrix system because of the erosion of the outer or the front layer of the delivery system, and slowly it will release. So the degradation can be controlled by increasing or decreasing the concentration of polymer which we have used to prepare this delivery system. Okay, now and second phenomena is the physical erosion. So physical erosion, you can see the very first, if you see the first one, this is the drug containing polymer matrix. So what is going on here? If it will come into the contact with the water, what will happen? The first forefront, this is the first forefront, this layer, it will cause the surface erosion. Okay, and afterwards the bulk erosion. So when the bulk erosion will occur, the ultimate the entire mechanism will dissolve and it will release the drug over that particular site. Okay, so this is how we are controlling the release mechanism, by controlling the physical erosion by blending a suitable polymer in a optimized ratio. Okay, so both this cavitation and this porosity are the responsible for this particular uh, release mechanism. Now, another one is the diffusion control system. So generally, we have to study this fixed law of diffusion, which you can see this is nothing but a concentration gradient. So this concentration gradient, this diffusion phenomena, you all have seen. I will give you one example. Like if you take a glass of water, okay, and if you add a tea bag, so what will happen? This tea bag inside drug is there. So what will happen? You can see the diffusion through this polymer. Drug is coming out, and the entire clear water will change into brown color. Have you seen it? Have you seen this phenomena? Or you have observed this phenomena? Okay, so yes, so this is what the diffusion process is all about. Okay, so this particular diffusion process is responsible to release the same drug from the entire region to the bulk solution of the glass. And why it is releasing? Because concentration here is too high inside this particular system, and concentration earlier in the bulk was very low. So since there is a difference between the concentration, so the rate of diffusion will be high. Okay, after some time, when the color will change to brown, you will see and you will observe, now there will be a condition, there will be a time when there will be equal concentration between the bulk and the inside cavity. So in this condition, there will not be any flow of concentration, or there will not be any flow of drug from this matrix. Okay, so this is the thing we have to consider in the fixed law of diffusion. So this particular case, by considering this, we need to develop a delivery system which should not dissolve, but it will form a diffusion layer, so that the inside drug molecule should diffuse from the internal core towards the bulk solution in a sustained or controlled release manner. Okay. So this is, as we discussed in the earlier slide, this is the drug release from the polymeric matrix or the reservoir-like system. And here you can see the degradation, sorry, not the degradation, the diffusion. Okay, because it will not dissolve, it will release the drug. I will show you one example as well. This is a drug release mechanism for the polymer carbopol. It will not dissolve in the water, but yes, carbopol will draw the water and it will get swelled up. So we are generally measuring the swelling index of this particular polymer. So to release the drug in a diffusion manner over a long period of time, what we can do is we can play with the concentration of this particular polymer, so that it will not dissolve afterwards, but it will swell, and from this swelling only it will have a good amount of concentration inside the tablet, and over a long period of time it will slowly release the drug up to a certain period of time.
Drug is dispersed homogeneously throughout the polymer matrix tablet. Drug release from tablets with carbopol polymers or Novion polycarbophiles is controlled by drug diffusion through the gel layer that the polymers form in contact with aqueous medium. When the carbopol-containing tablets are placed in contact with dissolution medium, the following occurs. The drug in the outside layer of the tablet exposed to the bathing solution is dissolved and then diffuses out of the matrix. The polymer swells fast to form a hydrated matrix layer, hydrogel. Due to the cross-linked nature of the polymers, the hydrogel is not single entangled chains of polymers, as is the case with linear polymers, but discrete microgels made up of many polymer particles in which the drug is dispersed. Carbopol polymers and Novion polycarbophiles do not dissolve in aqueous medium, rather they swell. In contrast, linear controlled release polymers such as hydroxypropyl methyl cellulose and hydroxypropyl cellulose are water soluble because they are not chemically cross-linked. The hydrated matrix layer controls water penetration into the non-hydrated core and diffusion of the drug through the hydrated matrix. Unlike linear polymers, Carbopol polymers and Novion polycarbophils do not dissolve during the release process.
So this is also a kind of phenomena by which the drug can be released over a longer period of time to control its release mechanism. Okay. So diffusion control release system. So diffusion can also be controlled by using a this polymer membrane, which you can see. Okay. So this will form a thick hydrodynamic thick layer around that polymer, and it will release the drug up to a longer period of time. So it is these particular things are generally a reservoir-like system, wherein the drug is there in the center core of the delivery matrix, and due to the presence of the outer layer, it will it will not get degraded, but yes, it will swell and it will form a diffusion layer, and from that particular thing, that reservoir, the drug will release up to the longer period of time. So this is again another phenomena by which there are the solvent activated systems are there. So these solvent activated systems, what generally they supposed to do is, you can see here the inner cavity, this particular entire region is the drug solution. Okay, so we are seeing it generally in the form of implants. So implants, what we can do is it can be refilled as well. Okay, by injection we can again refill it. So you can see here osmogens. So these are the particular bag or the membrane which upon in contact with this water it will swell. The moment it will swell, it will squeeze the internal this collapsible tube, and once it will be squeezed, the drug will release from the outer membrane. So you can see, to control the rate of release of the drug, this particular outer structure of the implant, from here you can see this face is basically an impermeable polymeric membrane, and rest of the body from this side to this side is a semi-permeable polymeric membrane. Again, to control the release up to a longer period of time, I can reduce the semi-permeable and I can increase the impermeable membrane up to this point. Okay, so again what I can do, I can control it, the release. The lesser I will give the semi-permeable exposure, the drug will be released up to a longer period of time. Okay, so you can see example as well. This is the osmotic pump. It can be planted inside the body by a small surgery, so that through this pump mechanism, the drug can be released and it can be maintained in the body of a patient up to a longer duration of time. Not for the hours, not for the days, not for the weeks, but yes, it can be used for months and years also. And after that, when the drug release has been stopped, it can be again, it need to be operated, and this pump can be refilled again. Okay, let's see. This is the
Alzed osmotic pump
osmotic pump. It does not require a
have no
battery.
batteries, no electronics, and no moving parts that can fail. The pumps are self-powered through the reliable process of osmotic displacement. Here's how it works. Alzed osmotic pumps consist of 3 concentric layers: the inner drug reservoir, the osmotic layer, and the outer rate-controlling semi-permeable membrane. The reservoir within the core of the pump is filled with the test solution to be delivered. When the pump is implanted, water is absorbed through the outer membrane. The water entering the pump expands the osmotic layer, compressing the flexible impermeable reservoir. This enables the test solution to be released through the exit port at a controlled, predetermined rate.
So I hope now you can understand this. So to control the release up to a longer period of time, what we can control is the surface of this particular outer membrane. If you give maximum exposure to the semi-permeable membrane, and if you give the low exposure of the semi-permeable membrane, it will extend the release mechanism up to a longer period of time. Okay, This is the biodegradable systems. Okay, so basically this biodegradable system, this should be of high biocompatible also. As the name suggests, it's a biodegradable system. That means after the preparation of a delivery system, it should get degraded inside the body. Okay, so these biodegradable systems follow all three mechanisms of drug release, that is the surface or bulk erosion, met polymeric matrix, as well as bound by enzymes, hydrolysis, and diffusion. But at the same time, it is also having a good tendency to solubilize, and also it should be uh, biocompatible and degradable easily with the body. So there are certain wafers which can be used to to suppress the growth of the tumor, because if patient is having a brain tumor, okay, and after surgery it has to be removed. So after this, what the next thing we can do is, just after the surgery, since it is a brain region, it's very critical also. You cannot do the surgery very frequently into this organ. So what after the removal of the tumor, you have to assure that there should not be any cell proliferation afterwards. So to avoid those cell proliferation, there should be a mechanism, or there should be delivery system which can be placed in that particular tissue, and afterwards, after a certain period of time, it should release the drug also. And also, as I told you, you cannot open this organ again and again. So that's why there should be a mechanism which can release the drug up to years, so that no other cells, cancerous cells, can grow up there. Okay, so such kind of delivery systems are there, and based on the need, there are certain kind of development of those delivery system. I will show you another video by which you can get an idea what is the polymeric drug delivery systems has been developed by different engineering techniques. Okay. Whatever we have discussed, you can see here in this video.
A wide variety of polymeric biomaterials are components of the various formulations and devices that are routinely used for delivering drugs to the body. This video reviews the current, state-of-the-art drug delivery systems that release drugs in a controlled manner. When a drug is released, the blood plasma level rapidly rises, followed by an exponential.
This is the frequency of the dose, because initially, as you can see, the x-axis is the time, another is the drug level. So there are two windows you can see here, first window, and here is the second window. Okay, so this is the maximum desired level. So your drug, whenever it is reaching up to within this window, you are getting a response. So you can see first dose. This is the first dose. The dose reaches its maximum and it declines. Okay, now as per the doctor suggestion, the patient has been told that you have to take here second medicine. So the moment he took second medicine, again the drug level in the body will rise, and again it will decline. So again the third dose is required to maintain this therapeutic window, because this time here, this particular blank space, you can see patient is not at all having a relief. Okay, like a general, take an example. If some pain is there in your body, so you are taking diclofenac sodium. So diclofenac sodium, if you take one dose, it will give you little bit pain up to certain period of time. Afterwards, again the pain will start. Why? There is a decline of the concentration of the drug. Now it is not available in your body. So again you have to take the second dose. Then it will give some relief up to certain period, and again you have to take the third dose. So to minimize these particular three doses, we need to develop a sustained release formulation. That is why diclofenac sustained release formulation is available, diclofenac SR. If you take that tablet, it will come within this therapeutic window, and it will release the drug up to maximum period of time. And uh, rather than taking 4 times a day, you can take SR tablet, sustained release tablet, once, or max to max twice, or once is also sufficient enough to get the relief over a longer period of time.
decay as the drug is metabolized and or eliminated from the body. The figure shows drug concentrations above which the drug produces undesirable side effects, below which the drug is often poor.
So this is the thing, you can reduce the,
For these reasons, there has been a great interest in developing controlled release formulations and devices that can maintain the minimum effective
drug level can be maintained very well.
Four examples of these systems are
These are the things:
diffusion control, water penetration control,
already have seen,
water penetration controlled drug delivery systems, and magnetic drug release system.
going to take
it's
it's already we have done.
Types of drug use of the agent compartment surrounds
the chemical
I will go directly to this professor
of the Massachusetts Institute of Technology had this to say.
Short in the form of the formulation research. His name is Roger, and he is from the Massachusetts. And what they are working is, they are working on the different kind of polymeric materials to release the drug up to a longer period of time. And particularly, their team have developed one wafer kind of delivery system where the anti-cancer drug has been loaded up, and those wafers can be implanted into the brain surgery, into the patients where they have just now removed the tumor, and they will place that wafer into the brain or that operated region, and they will place this wafer and it will release the anti-cancer drug in a slow manner, not it will be a shooted up kind of thing, and into release manner up to 3 years of time, so that there will not be any growth of the tumor after surgery. Okay, just listen to his these things.
Various mental health diseases like schizophrenia, many heart disease, many others.
This is the wafer.
So let me take brain cancer as an example. What we did there is we created a polymer, and it's a very special kind of polymer, plastic, that just dissolves layer by layer, and so it can't dump the drug out. And also, what's good about it is by changing the chemistry, we can actually make the polymer last anywhere from a day to 3 years or any time in between. And what happens now in this treatment is like the neurosurgeon would operate on the patient, because they're going to do that anyhow, and then they take out as much of the tumor as they can. But before they close the patient up, what they do is they put these little wafers in. This is an example of one of the wafers. And usually they put 7 or 8 in right at the site where they operated. And then the idea is the wafer keeps releasing the drug over, say, a month or 2. In the meantime, the wafer dissolves completely, so it's not in the brain. And the idea is the wafer will deliver the drug locally right to the tumor, killing it, but also it doesn't get to the rest of the body, where it could cause a lot of side effects.
Okay, so this was the application of such kind of strategies, wherein we have seen just now the patient who was operated with the brain surgery. So after the post-surgery management, the wafers have been placed. They are prepared from different kind of plastic polymers, which are prepared to be diffused layer by layer. Okay, initially first layer will diffuse, and along with the diffusion or erosion, the drug will release, then second layer will release, then third layer, like this. So they have prepared it layer by layer, and it will release to the extended period of time. So this is how they have done. And also, since these anti-cancer drugs are very kind of not suitable for every host cell inside the body as well, so by giving a localized action, its toxicity is well maintained, and also they have prevented the unnecessarily movement of this anti-cancer drug inside the entire body. So this is a localized effect. So this is how there is a development, and these are the certain kind of polymers which are used to control the release systems. As I told you, there are natural polymers and synthetic polymers broadly, and also there are branded polymers as well, which are readily available, and these particular things are patented. And these 3 basic categories are there. If you see the natural polymers, so basically the source are from the protein based polymers and the carbohydrate based polymers like protein-based polymer, collagen, albumin, gelatin, and carbohydrate-based polymers are alginate, cyclodextrin, chitosan, dextrose, agarose, hyaluronic acid, and cellulose. So the cellulose are having certain derivatives as well, like ester and ether derivatives that can be used for the formulation of the delivery system. Apart from the natural, synthetic polymers are also there, like polylactic acid, polyhydroxy butyrate, and polycaprolactone, and polylactic acid, and you can see other examples as well. So polyvinyl alcohol, polyacrylic acid, polyethylene glycol we are generally using because it is having a hydrophilic chains as well. So if we bind this particular polymer around any nanoparticle, so it will provide it a stealth kind of nature. It can be very well taken up by the RES, not by the RES system. The RES system of the body will not detect the presence of this nanoparticle because of the surrounding of the nanoparticle is done by the hydrophilic chains of polyethylene glycol and polyanhydride as well, orthoesters, diblock copolymers, and triblock copolymer. These are the certain block copolymers which are used to you know to control the release behavior, like diblock copolymer, polylactic co-glycolic acid. So it is a combination of polylactic and glycolic acid. So in a different ratio, we can take this both the polymers for the delivery system. Trico block polymer, like you can see the PEG based. If you see the example of PCl, polycaprolactone. So if you are preparing a thermosensitive polymer, so PCl, PEG, PCl. So you can use these three trico block polymers, in which you can use two hydrophilic chains at the end, and middle section you can keep a hydrophobic one, so that it can be triggered by some external environment, and it can be converted from solution to gel. So this is a smart kind of delivery system. And other branded polymers include this interact polymers and phenyl soluble enhancing polymer, ethosols and methosols. These are some polymers. We will see the application of this particular polymers. So on the basis of this their pattern and their degradability, these polymers are classified into basically two kind of things, like biodegradable polymers and non-biodegradable polymers. So biodegradable polymers are generally classified into three categories: natural, synthetic, and semi-synthetic. And non-biodegradable are classified into two categories: synthetic and semi-synthetic. So natural, we already discussed the category, and this is the particular polymers which are used in the as a, for the pharmaceuticals polymer, and based on their origin and their nature of degradability, they have been broadly classified. Okay, if you see the natural polymers, these are of high compatibility and biodegradable in nature. So this include the origin, like these are animal origin. Here you can see the animal origin, plant origin, microbial origin, and marine origin. So these are some examples of these natural polymers from different sources. Okay, and the best part of using these natural polymers are again, they are of not that much expensive, as well as they are highly biocompatible. So another one is the collagen. So collagen, you can see the application, like the ocular particles of cyclosporine, then ocular inserts, then ocular gels, ocular shields and films, collagen conjugates, aqueous injections, monolithic devices, and microsponges. So you can see maximum these collagens are that much compatible with the environment as well, and most of the formulation you can see these are for the ophthalmic preparations, which can be used to deliver the drug into the eye. So these are of this should be of high quality, and this should be of high particular compatible as well. So another example is the albumin. You can see there are certain examples wherein the drug is being used with the albumin nanoparticles, and it forms a good complex as well with the albumin. So because albumin is responsible, this is to bind with the drug, and basically you can see there are the drug albumin nanoparticles available, drug albumin conjugates are available, drug albumin ligand and antibodies complexes are there, and research have been done on the basis of drug albumin and complexes like paclitaxel and albumin nanoparticle is prepared, and there are certain other example you can see around for the preparation of nanoparticles by using the albumin as a polymer. Okay. Now, gelatin, as you all aware of the capsule formulation, so gelatins are used since a long time, and because of its high compatibility and its availability as well. So earlier it was the hard and soft gelatin capsule. So hard gelatin capsules are those capsules where we are filling up the granules or the powders directly into the capsule. Soft gelatin are the capsules which you can see some liquid preparations are filled up into this kind of capsule. So these are known as the soft gelatin capsules. Okay, a number of capsules are available in the market for under both the categories. Now you can also see gelatin nanocomplexes. So there are examples are cisplatin loaded polymer gelatin nanocomplexes, then amphotericin loaded lipid gelatin complexes. So the complex has been prepared by using the different kind of drugs which are hydrophobic in nature, and they are formed complex with the gelatin. Now the gelatin surface has been also modified with the formulation to provide a stealth gelation formulation. So you can see I told you earlier also the term stealth generally it only comes when it has been surrounded by the pegylation. So again it has been pegylated, and the drug used was the doxorubicin. And by the surface modification of the gelatin, this has been prepared, and to coat the nanoparticle with the pegylation, the gelatin has been used and the pegylation was done, and that's how they have made it stealth in nature. And also the modified formulation with high efficiency gelatin based on the targeted delivery. And yes, there are also formulations of micro and nano particles as well, and these you can see for the delivery of the protein and the drug delivery, including gene and anti-cancer drug delivery, ocular drug delivery, enzyme immobilization. So these are the applications of the gelatin. So here I have taken few examples for the alginates polymer also. So basically there is a vast variety of tablets you can see with the form with the alginate, like the matrix tablet, floating tablet, microsphere, raft, beads, and calcium alginate nanoparticles and ocular gels are available, which in which the research have been done, and they have used alginate to form the kind of delivery mechanism, delivery systems which can be used to control the release of the drug over a sustained period of time. And also cyclodextrins, you can see cyclodextrins are generally a cavity filled up with the hydrophilic chains around. So any hydrophobic compound which is not soluble can be placed very well in such kind of, can be included into such kind of cavities, and ultimately after the formulation the entire cavity as it is surrounded by the hydrophilic ends, so its solubility of that particular hydrophobic compound can be increased by incorporating into the cyclodextrin complex. So ultimately you can see the formulations are prepared for the oral delivery of such drugs which are not soluble, and further what happened is its solubility got increased, and as the solubility got increased the absorption become very good, and after this the bioavailability will also increase. Okay, apart from that there is a rectal delivery, nasal delivery, transdermal delivery, ocular delivery, control and targeted delivery. Okay, and also the delivery of protein and peptide has also been observed to increase its permeation through the blood brain barrier. So these are the different use of the cyclodextrin polymers, and based on the different kind of route of administration. Now, chitosan is also a very excellent class of polymers which have been taken by many scientists, and still the research is going on, and it provides a versatility in terms of different properties which can be used and which can be deployed for certain kind of delivery systems. Like you can see the formulation with Catterson derivatives, um, macrospheres have been prepared, and these particular macrospheres can be prepared by many methods, like spray drying method is there, and also ion gelation method is there, by which we can prepare the Catterson macrosphere. And the drug used here was the insulin, and it was the oral delivery. For the oral delivery, it has been prepared. And low molecular weight alkylated chitosans is used for the uh, non-viral vector for gene. These were the formulations. Apart from this, there are certain kind of formulations like matrix tablet, hydrogel, and certain nanoparticles which are prepared by the catoside and combination of sodium alginate. And also indomethacin has been loaded into ocular this kind of formulations where the chitosan is involved for the delivery of indomethacin through uh, eye route, that is the ocular delivery. Okay, so they nano emulsions and nanoparticles can be formed wherein we can use these particular things where a patient has been operated, some eye surgery is there. So after that, to reduce the pain, such kind of delivery system can be instilled into the eye of the patient into the form of drops, and the patient will be feel comfort over a longer period of time because of the chitosan. So this presence of chitosan can be very well taken up for the release, for controlling the release behavior of the drug for the longer period of time. So, likewise, this is the example of hyaluronic acid. You can see the vast application of this particular drug also in the field of ocular delivery, film and microspheres, nanospheres, and you can also see it has been used for the parental formulations as well, and nano carriers as well. And there are other different kind of research has been done by using the hyaluronic acid. It is also been used for the bone regeneration and tissue regeneration as well. And apart from this, the cellulose are also very important, and you can see its application in the pharmaceuticals in the field of bioadhesive and mucoadhesive, film coating, enteric coating, and these are cellulose derivatives which we can use for delaying the release of the drug. Okay, suppose like I will tell you this enteric coating, it is a coating done on the tablet, because if a normal tablet, if you plot a graph, so it is a time, it is a drug release. So in the case of normal tablet, it will go and it will slowly come down. That means within this margin only, suppose it is a 1 hour time only, the drug will go to its maximum concentration and again it will decline. Okay, but in case of enteric coating, so this particular coating is done over the tablet to prevent it disintegrate into the gastric fluid, and in the gastric fluid the drug will be there up to 2 hours. So we don't want any release of the drug up to 2 hours. Okay, but we want it to release in the basic environment, so that whatever the kind of drug we are using, if it is not at all compatible with the gastric fluid, if it will degrade in the gastric fluid, we need to secure it. So to secure it, we need to coat the tablet in such a manner that it should not give any release. So its release profile will be, it will go like this: 0, nothing will happen. After 1 hour, nothing will happen. During 2 hours or 1 or between 1 to 2 hours, it will give a release. Okay, so what we have done is we have delayed it up to 2 hours. So by this time, the stomach will be this tablet will be moved from stomach to the intestine, and it will release the drug into the basic pH. So this is the strategy for enteric coating. And also you can see the designing of the extended release formulation, diffusible matrix, and semi-permeable membranes, which we have seen in the osmotic pumps are used, which are prepared by the cellulose. And also there is a vast variety of PLA, polylactic acids, both into the food industry it has been used. Okay, and there is a vast variety in the field of medical industries also, for implants, preparation of implants, screw, rods, pins, mesh, etc. And these are highly biodegradable, the biodegradable containers also, and storage tanks have been prepared by the use of the PLA. And PCL, caprylactone, so you can see there is a different kind of formulations which has been prepared by using the PCL, like micelles, hydrogels, films, fibers, scaffolds, and nanoparticles. So these are the different area where the polycaprylactone is being used for the preparation of delivery system. And apart from this, polyvinyl pyrrolidine is also used, and it is basically used to prepare the nanoparticles and blood plasma expander, soft contact lenses, coating material, stabilizers, bioavailability enhancer, drug release enhancer, binders and adhesive, film former, test masking agents. So you can see the vast variety of applications are available. Some of the PVA formulations are marketed as well, like copolymers, you can see ophthalmics, tablets, implants, and transdermal topicals. Okay, so by seeing the video and by mapping it, you can now understand what are implants, what are transdermal topical patches, tablets. You know very well. Ophthalmics are the eye drops which are used. So in all, you can see there is a involvement of PVA, and these particular PVA are so biocompatible that they passes all the test and regulatory guidelines, and they are into the market, showing the efficacy of these particular delivery systems, which can be able to use or able to release the drug in a controlled release manner. Apart from this, these are the branded polymers which are used, like amberlite, which are used for the sustain release pattern of the drug, taste masking, and tablet disintegrating agent can be used, and also to mask of the bitter taste of the tablet. Okay, this particular polymers are used. And ethosols and methocells, as we have seen, these are the branded one. So this ethocell having a application in the food and the feed industry and the pharmaceuticals, cosmetic industries, conductive, paste, print, and ink, and also into the form of ceramics. So ceramics, basically the rheological uh, modifier, providing the green strength to the uh, un unfurred ceramics. Okay, so ceramics is also playing a very important role as a biomaterial to be used in the body, because it prevent the microbial growth. So these ceramics are very well taken up in kind of implants and all. Okay, so let's see, there are different kind of materials, okay, which we can use inside the body. So these particular materials are broadly divided into different groups based on their crystal structure, bonding, and macrostructure. Okay, so on the basis of the bonding, if you see on the basis of bonding, these materials are divided into metals, ceramics, and polymers. Okay, so what happened is, the material that bonded via metallic bonds are called the metals. Okay, so these are the materials which are bonded by the metallic bonds, and they are having abundant source of free electrons. And because of these free electrons, they are having good thermal as well as electrical conductivity. Okay, both thermal and electrical conductivity they will show very good, and they are also malleable also. And they are having excellent mechanical strength. And those materials which are primarily ionic or covalently bond are called the ceramics. Okay, but these don't have any free electrons over their surface, and that's why they are a very poor conductor of electricity. Okay, and they are both thermally and electrically they are very much poor conductor. Okay, so this is the second class. The third class belongs to the polymers, and due to the covalent bonding, most polymers are, again, they are non-conductive. And if you combine all those three class of material, the metal, ceramic, and polymer, So when these three materials are mixed together without losing their inherent characteristics, they are designated as the composites. Okay, so this is also widely applied in the human body, and I'm speaking on behalf of the pharmaceutical, so that's why I will be taking only those examples which are there which can be used up into the some kind of or some kind of disorder, or which can be placed within the body of the patient. So apart from this, the metals are having a vast variety of metals, ceramics, vast variety of ceramic. Apart from the this pharmaceuticals, if you take examples, there are so many engineering involved into the fabrication of the materials by the use of these kind of materials, which is belongs to the class of metal, ceramics, and polymers. But we are focusing on those materials which are biocompatible, and that's why I will take the name as well as the biomaterials, so that it can be very much compatible with the body system. So those types of materials can be divided into some synthetic and natural sources. We have seen porous materials and dense material on the basis of their crystallinity, semi-crystallinity, and amorphous nature. They have been divided, and these particular materials are divided onto their classes, like we already discussed now: metals, polymers, ceramics, and composites. So this is the particular classification of the different kind of material. Okay, so these range of materials that are employed to control the release of the drug and other active agents. So these particular properties are like elasticity. Okay, so these elasticity, like you can see, the polymer, the polyurethanes are responsible for the elasticity. So selection of these biomaterials for any kind of delivery systems were based upon their inherent property. So polyurethane is generally used for the known for the good elasticity. So likewise, for the insulating ability, polysiloxanes and silicones are used. For physical strength and transparency, polymethyl methacrylate is used. And for hydrophilicity and strength, polyvinyl alcohol is used. For toughness and lack of swelling, polyethylenes are used. And for suspension capabilities, polyvinyl pyrrolidines are used. And apart from that, for the control release mechanism, there are certain class of materials which are designed to get degraded inside the body itself, like polylactic acid, which is generally known as PLA, glycolides, that is PGA, polylactic coclides, that is the PLGA, and polyanhydrides and orthoesters. So these are the class of polymers which are intended for the medical applications, and also they are having an excellent ability to get degraded into the body. Okay, so this, when we are talking about the biomaterials, so these particular biomaterials are should be uh, able and can be used as a medical applications to perform a body function or to replace a body part or a tissue. So basically our tissue is divided into two parts, that is the hard tissue and soft tissue. So if you take an example of hard tissue, bone is there. So bone regeneration you might have heard, and tissue regeneration is also there. So research is going on based on those two areas broadly. If I give an example, like a scaffold, if you see, so tissue engineering is basically divided in, this is the scaffold, is the platform for both bone regeneration and tissue regeneration. So to have such kind of regeneration, high compatible materials are required, and that's why these biomaterials are that to be in picture, and they must be of high biocompatibility both in cellular and systemic level. So these biomaterials are used to repair and replace any tissue, organ, or function of the body. And some of the major advances in the materials with the synthetic polymers such as Teflon has been noticed in case of vascular grafting and surgical application. Also, hydrogels are used to for the invention of the soft contact lenses. and polylactic glycolic acid (PLGA) was developed as a resorbable sutures, and ceramics such as the calcium phosphate are used as a synthetic bone analog. And there are two classes of those things like autograft and allograft. Autografts are the bone taken from other sources of the patient's body, and allograft are the processes of the cadaver bones. And along with that, there are certain alloys which are used of high compatibility, and like the titanium alloy, which can be used for the bone replacement in case of knee replacement in the orthopedic implants, and they are also having a lighter weight. So these particular titanium alloys are used in case of the knee replacement to the elderly patient where they are suffering from knee pain and knee joints. And apart from this, ceramics are used for the dental application also. I will show you a few examples where we are using this biomaterial inside the body. I'll give you example of bone replacement, knee replacement. Here you can see. So you can see this knee. It is its internal particular surface is being ruptured over a long period of time when the patient's grown up into their old age. So there is a deformity in the bones has been observed, and basically the accumulation of the uric acid and erosion, they are the nerve ending of those bones. When they will come in contact with each other, they rub, and as a result of this rubbing and this abrasion, the pain is induced. So these things can be overcome by placing a certain kind of compatible biomaterial which can provide sufficient strength, and also at the same time it should not cause any kind of immunity disorder to the patient, and also it should be of high compatibility. So let's have a look on the procedures by which they are placing the metal, like biomaterial, for example titanium alloy, uh, to this this patient who is suffering from the who is looking for the knee replacement. Okay.
The knee consists of the tibia or shin bone, femur or thigh bone, and patella or kneecap. In knee arthritis, the cartilage on the end of the bones slowly wears away, causing pain and irritation, making daily activities difficult. Arthritis can affect all or just one area of the knee. In this case, the entire knee is affected and a total knee replacement is performed. During the surgery, your doctor uses a series of guides to help remove the damaged bone and prepare the femur, ensuring proper implant size and alignment. Next, your doctor will prepare the tibia using a series of guides to ensure proper implant size and alignment. The final tibia preparation is completed and a trial spacer is used to confirm knee stability. Next, your doctor prepares the patella to accept the implant. A trial implant is placed on the patella and the knee is checked for proper implant size and stability. The trial implants are then removed and the surfaces are prepared for placement of the final implants. Your doctor applies bone cement on the backs of the tibia, femur, and patella implants for final fixation. This completes total knee replacement.
Another example is the dental surgery. I will not take much time. I will just forward this. So you can see space is there, gap is there, and it has to be one. Teeth has to be placed here. If you cap it with a crown, so after a long period of time, when the patient is chewing food, it will cause some pain also. This cap will give sensation. So to avoid this, the option is to use this kind of implants. By surgery, it can be placed over. So this is a particular process. Scanning, first scanning will be done, and they will map it, and they will fabricate this particular implant, and they will fix it with the other part, and afterward the crowning can be done. So this is the scanning process, and you can see here the parts, the implants which will be fixed in the upper portion, and the abutment will be fixed with the implant, and after that it will be crowned up with this crown. So this is the process, and you can see it has to be fixed like this, and above it the crown is coming. So if you see this particular thing which has been placed inside the this tissue, it should be of high compatibility, and also this should be well taken up by the surrounding tissues also, and there should not be any microbial growth once it has been placed. So basically the ceramics are used, and the tissue will cover up the entire region, and the healing process will be done. So this is the thing. So you can see. So this is a side view. So after this drilling, this implant will be placed here. And now next the healing will be done. So healing is a it will automatically start, and this biomaterial should be well taken up by the body as well. So these are some application area wherein these biomaterials are used apart from the control delivery systems, because we have to cover a class of different kind of polymers as well, which are used not only to deliver the drug, but how we can see that they are used in different aspects inside the body. And yes, to use those kind of biomaterials for control release mechanism and for other applications, a high end of characterization is required. And you can see here these all are the systems which can be used based on the three classes we have discussed, that is the metal and the polymers and the ceramics. So you can see orthopedic implants, stents, dental implants, fracture management devices are coming under the metal category, and breast implants, ophthalmic devices, sutures, skin graft are coming under the category of polymers. And as well as the ceramic, we have seen bone graft, heart valve, dental implants, and pacemaker is a combination of all those three, like metals, some portion of polymers, and ceramics has been used. So to select this kind of biomaterial and to select the integrity of this biomaterial, there are different kinds of spectroscopy or some characterization techniques are involved. For example, you can see the IR spectroscopy, Raman spectroscopy, X-ray, photoelectron spectroscopy, ultraviolet visible, nuclear magnetic resonance, and mass spectrometries are available. These are the different characterization techniques by which we can select our biomaterials onto the host tissue and the living systems, so that no immunological interaction can be observed. So there will not be, after that, if you select it wisely, there will not be any kind of device failure in the patient. So uh, these are some different kind of characterization techniques to select our biomaterials. Like the crystalline properties can be seen by using the thermogravimetry, DTA, and XRD analysis. Particle size can be characterized by using the particle size distribution, dynamic light scattering, and small angle scattering systems. And apart from this, surface and chemical analysis can be monitored by zeta potential, contact angle, and EXRD. And apart from that, the vibrational spectroscopy you can see, under which there is a Raman spectroscopy and Fourier transformed infrared spectroscopy (FTIR), and nuclear magnetic resonance, that is NMR. So you can see a broad variety of characterization techniques are available to select a different class of your biomaterial. If you are looking for only the morphology, if you're interested to know the morphology of the polymer or morphology of your material, you are having a certain kind of methods, microscopy methods, by which you can see whether your formed or fabricated material belongs to that class or not, what you have thought of. So like for morphology, you can see SEM and TEM. Basically it is being used to see the morphology of the structure. So based on the quality and the application, we are categorizing our biomaterials. And during the formulation, a formulator is knowing that okay, my formulation should be in nano size. So for nano size, I need to check the particle size first of all. So I will go with the DLS properties, and I will see the particle size. Upon the upon knowing the particle size, if it belongs to the nano range, okay, then my next task will be checking its compatibility with the drug, whether this polymer is compatible with the drug or not. Okay, then afterwards I will be looking for the morphology by using the SEM and TEM, so that I can see whether a pores has been created in the surface or not. So SEM can give me an exact idea about the surface particular, but if I want to know the internal structure as well, I have to go for the transmission electron microscopy. So based on your requirement and your need, you can opt any of those techniques for your research work if you want to pursue in this area. So there are other few examples. The same we have seen in the previous one, and this is the same version of that particular table, where there are they have divided into the structural and chemical analysis as well, and morphology to check the outer surface of your delivery system. Okay, so with this I will end this session, and these are the references which I have used to prepare my slides. And yes, thank you very much. If you have any questions, please, I will be happy to answer those.
Thank you very much, sir. Thank you very much for delivering this session with so much information related to controlled release system, drug delivery system, then polymeric materials, Alzheimer, cancer, focusing all the recent research trends of biomedicals. And thanks for those animated videos also, because every people able to learn many things related to the techniques and the technology. Thank you, sir. Now the session is open for interaction. If any participants have anything to know from sir or ask sir, they can. There is a question at the chat box. Is hydrolinic acid is used as alternative of synovial fluid?
See, those research, has been, going on, and yes, if it is a kind of some no immunological response is not there, then certainly, based on its biocompatibility, it can be used up, and Basically, the purpose of this particular presentation was to, maybe somebody is looking to prepare some nano formulation, somebody is looking to go for some kind of implants and some other biomaterials which they are planning to do that. The first and the foremost thing you need to start is the screening of your biomaterial. Okay, so because there are certain class of polymers which will be there, but if it will not give you the required release profile, drug delivery, or if you are selecting a particular polymer for the targeting as a targeting moiety. Suppose you prepared a nanoparticle with a different class of polymer, and you are targeting it with putting a ligand over the surface of this nanoparticle, and you have to be very much sure that it should not bind with other overexpressed cellular surfaces. So you need to deploy certain modification into your chemistry of your structure of the particular particle, so that it can bind exactly the same targeted area. And yes, it should be very much biocompatible, and you can go through the literature as well if you find something like whether it can be used or not, because I am not that much aware of that particular hyaluronic acid application.
Dr. Tharmalingam has some queries. He raised his.
Yes, yes, please. Yes, what is the query?
Another question
Yes.
from Dr. S. Selvam. Titanium alloys, stainless steel, and polymers like PE, PIC, etc. are used for orthopedic application. Which one is best for orthopedic implants?
See, there is a, why I'm focusing on biocompatibility, you can check on the Google as well, GBS syndrome. Okay, so this GBS syndrome, generally it's a very rare kind of immunological disorder wherein your body will, act against your immunity. So these kind of systems developed in certain patients when this knee replacement has been done. So it first of all, during the consultation, a thorough history check of the patient should be done. Yes. Second part is based on the different classes of material, why alloys are better. So obviously, the first thing is the strength. Your biomaterial should be malleable enough, it should be tough enough, it should be biocompatible, it should be, the most important is the weight, it should be light enough. So if it is based on these different classes of material, these titanium alloy are because of their lightweight, they are the most preferred one. And also there are certain kind of limbs, prosthetic limbs also, which are not implanted inside the body, are also prepared uh, by varying with different certain properties. So yes, light weight of the metal is more preferred, and compatibility as well for the selection of the material for the orthopedic surgeries. Its weight should not be more actually.
Is there any more questions?
I have one question.
Email address, may I share that email address?
I have one
Yes,
question
yes, sir.
to the audience. Whether because it's a mixed class of domains, so whatever the presentation I have given, whether it is understood to all or not. I want to see. Because these pharmaceuticals are very vast actually, and that's why very particularly I have selected these slides. Before putting the content also, I was thinking that okay, it might not be a boring class, so that's why, to show you the better picture of that theoretical aspects, I have taken few example of a video so that we can get some understanding. I know that it's very difficult to get other domains area knowledge in one presentation. So, but I have tried something. But if it is okay, then yes. So it is a successful presentation for me also. Okay, thank you.
Thank you very much, sir. Thank you very much for sharing this informative session and giving us your valuable times. On behalf of CIPET CSTS Guwahati, thank you all, sir.
Thank you. Thank you so much. Thank you. Jai Hind.
Thank you. So dear participants, now tea break is there. At 11:45 we will meet again. At 11:45, talk will be delivered on the topic polymer matrix space core cell nanostructures for application in biomedical imaging. That will be the last technical session, and after lunch break we will have exam and the valedictory session. So I request everyone to be present in both the session, and you can be at the same line joining link, or you can rejoin again. Thank you. We'll resume again at 11:45. Thank you so much.
ঠিক আছে কেতিয়া কৰিম Very good morning to all the respected participants. So today's second topic will be delivered by Dr. Jutika Devi. Before I request her for her talk, I'd like to give a brief introduction, to put forward a brief introduction before you. Dr. Jutika Devi is currently working as an assistant professor at Central Institute of Petrochemical Engineering and Technology. She was an independent researcher and a scientist under WOSA scheme, Department of Science and Technology, Government of India. She had done her PhD in the domain of nanoelectronics for next generation biomedical and wireless communication devices. She was also associated with Indian Institute of Technology, Kanpur as a researcher, and she also received many prestigious awards. Among those, mainly like prestigious DFG fellow from German Research Foundation, then from DST, Department of Science and Technology in the Government of India. Also she got fellowships, different fellowships to carry out different research projects, and she completed different industry link research projects she did. She has 5 years of teaching experience in different postgraduate and PhD level students at Guwahati University. So prior to join in CIPET, she was associated with Guwahati University, Government of Assam. She has published many scientific journals with high citation index research papers she has published. And mainly her research papers were in the domain of microwave frequency for medical applications. She has published 1 book and 4 book chapters and delivered a number of invited talks in many seminars, national level seminars, as well as international level seminars, as well as in different webinars last 2 years. She is also associated with professional bodies like SPIE, the International Society for Optics and Photonics, International Association for Advanced Material, etc. Currently, she is working on core cell composites, nanoparticles for biomedical imaging applications. She is also recognized as an innovator by Worldwide Academic Industry Network on the focus area of communication and medical technology. And today, Dr. Devi will be presenting polymer matrix based core or and cell nanostructures for a specialized application in biomedical imaging. So I strongly believe that the research work of Dr. Jutika Devi will be presenting before you will definitely give you a path of thinking for how we use this nanostructure for biomedical applications, especially in the field of biomedical imaging. So with this short introduction of Dr. Jutika Devi, I would like to request Dr. Jutika Devi to kindly proceed for the today's talk. So over to Dr. Devi.
Thank you, sir. Thank you very much for this encouraging introduction. Now I am sharing. So today I am going to speak on polymer matrix-based core cell nanostructures for application in biomedical imaging. In the last 4 days, we have got an idea about what is polymer, how polymeric nanocomposites can be synthesized, how characterization can be done, and what are the applications, and from the material point of view, how we can apply it in different areas, basically in the medical technology sector. So in this talk, I am going to highlight briefly about the devices which are going to be manufactured, and some are already in the market for bioimaging and targeted drug delivery using core cell nanostructures. So we all know that nanotechnology is working on measuring, manipulating, and controlling at the atomic level. And the length scale is approximately up to 100 nanometer. And this size is the primary factor for which nanoparticles are different from microparticles. And another important point is surface area. As already in the last few lectures, we have got the idea how surface area for nanoparticle is different from the microparticle. And as we all know that surface area for nanoparticles are more compared to the volume. So that's why the unique properties, its unique physical properties are there. And for these unique chemical and physical properties, these nanoparticles can be utilized for different applications. Another important parameter is quantum size confinement. As we all know that the mechanical systems or macro bodies are governed by the classical mechanics. Then microparticles is there, and these nanoparticles are governed by the quantum mechanics. And in the quantum mechanics, since the size of the nanoparticles are comparable to the de Broglie wavelength, so Schrodinger's equation comes into play, and the particles shows wave behavior. And due to this, these nanomaterials become special for application in different optical devices. And as we all know that for metallic nanoparticles, color may change with size. So already from physical point and synthesis characterization of point, this is also a spatial parameter or spatial property, because earlier when we make LED, we use different type of materials for designing the light emitting diode. Now, if we use a single nanoparticle and if we can synthesize it by varying the particle size, then with the same material, by varying concentration, we can have different colors. So this also opens a new area of application for the nanoparticles. Another important thing is when we are designing a device, we always think about the market. So in case of nanotechnology also, though nanotechnology is not so much older in the commercialization, but nanotechnology impact many sectors of the global economy. And the global nanotechnology market is expected to achieve $33.7 billion by 2030 from $1.76 billion in 2020, as reported by the Market Survey of Global Nanoparticles. Semiconducting nanowires, carbon nanotubes, nanofibers, quantum dots, core cell nanoparticles, graphene, these things, these nanomaterials, when we are embedded in a polymer matrix, gives different type of potential behavior which make it extensively useful as a building blocks for different nanoscale devices. And if we go through the technology that are governing our daily life and all the things that are the electronics, and electronics remains one of the fastest growing end-use markets for nanocomposite driving robust gains. And polymer nanocomposites, as we all know, we can use, there is the speciality of polymer nanocomposite is the dispersion properties and the uniformity. And another important point is when we are using a polymer matrix are not so stiff and they're transparent. So this make it very useful for application in optical domain also. At very high frequency, we can use at near infrared region also, we can use this polymer nanocomposites for different applications. And this polymer nanocomposites, if we use a polymer matrix and the fillers, if we use semiconducting material, then also it can be used for different applications like photocatalytic, then photosynthesis, then for different solar cell, then for filtering properties. So for all these properties or all these applications, we can use semiconductor as the nanofillers and polymer as the matrix. And nanotechnology is the rapidly growing technology, and it has applications in different domain, and these are the key manufacturers of nano domain like Advanced Diamond Technologies, Advanced Nano Products Co Limited, Altair Nanotechnologies, Bruker Corporation, Chem Technology, Elitech Group, eSpin Technologies, Hanwha Solutions Chemical Corporation, Hybrid Plastics, and so on. And when if we Google, then also we will get that there are so many companies are there who are specifically synthesizing different type of nanomaterials of different shape, and that also they can measure research domain of the nanotechnology. And the market is also very good for this nanoparticle synthesization, as for different applications we need different size of application, and synthesizing with having all the properties, uniformity, the dispersiveness without agglomeration, it's a very sophisticated process. So many companies are based on it, and if we go to, then we are finding that different technologies are also there. And nanotechnology is considered as the next industrial revolution with enormous possibilities in many fields. But it is quite challenging that microparticles are there. We are making them nanoparticles. Then how their behavior or how the theory behind it can be modified, or will the same theory we can apply for those device design also. And cost is always a factor, because if we use metal nanoparticles, then always the cost matters. If we use, suppose, gold nanoparticles, if we use silver nanoparticles, then graphene is also very costly. But from application point of view, since tremendous applications are there, promising applications are there, so the world is investing lots of money, and the government agencies all over the world has developed multidisciplinary research and education centers to develop these nanoscience and nanotechnology based works. In India also, almost all the funding agencies has a specialized cell, like DST also. We have nanoscience and nanomission in DBT also, Department of Biotechnology also. And there are many institutions and organizations which works exclusively on these nanomaterials, like National Institute of Nanoscience and Technology, the Center for Soft and Nano Research are among them. And since nanotechnology applications are covering all the fields like agriculture and food, defense and security, energy sector, then energy harvesting also, as we have seen in our last 2-3 lectures, then optical communications, electronics, biomedical, biotechnology, cosmetics, paints, almost all, and in the food sectors also, in just like in the packaging also we used. Then for interconnection of the electronic technology also we use nanomaterials. And as predicted, this nanotechnology are the next semantic wave, because if we go through this figure, then we are getting that the years on which is governing, just like 1800 to 1853, textile brought the revolution. From 1853 to 1913, railway was there. In that way, it is expected that from 2025 to 2081, nanotechnology will lead the global market. And if we consider, everything is actually interrelated. So why nanomaterial and system parameters should we measure, and how? Then if we were for everything, synthesizing is also necessary. Proper choice of the material, how to synthesize it, then what are the characterization facilities. And if we go for biomedical sector, then there are many things which we have to take care of. Just like how can we predict whether the nanomaterial will remain stable in the human body? How can a nanomaterial be engineered to prevent unintended toxicity when introduced to a biological system? Then how the information environment will react with the nanomaterials, and how we can, in a controlled way, how we can put that nanomaterials to a human body. So a lot more things are there, and lot more research work is going on. So on that, suppose there are many properties, just like in our earlier lectures also, we have found that many chemical properties are there, many biological properties are there, there are many molecular dynamics are also there. Then many people has focused on the kinetics also. So in this study, there are another inherent properties of material, which is dielectric properties. So how these dielectric properties can affect the imaging techniques, or how these dielectric properties can be controlled for a polymer matrix based core cell nanostructures, and how we can predict the mobility and effective exposure of nanoscales chemicals. So in all these fields, research is going on, but there is a common kernel of questions and answers that would advance this research field and opens up on a multidisciplinary basis. That's why this nanotechnology and polymer nanocomposites, this area of almost all the branches of science and engineering are focusing on this area. So this polymer matrix-based nanostructure is a new class of advanced composite material. These questions now coming into mind. How, suppose for a bigger particle, suppose it is a microparticle, the theory which govern that microparticle, will the same theory will be followed for the nanoparticle, because nanoparticle is dominated by the quantum mechanics. So if quantum mechanics govern that, the wave-like behavior plays into the main role. So how we can change or how we can modify the properties, because for nanoparticles the main important parameters is size does matter. So since size matters for the nanoparticles, and the variation of sizes give us variation of sizes, variation of concentration, and different shapes give us different type of behavior for the same material. So there is a question how to scale down the parameters which are there, because we can't change the theory, but what we can do, we can scale down or scale up the parameters to make it fit for the nanomaterials. So till before over 150 years, we know that resistors, capacitors, and inductors were the three fundamental passive circuit elements. And passive lamp circuit elements are, we had earlier all the circuits which have lamp passive elements, just like resistors, inductors, capacitors. Now we are in the embedded technology. So all are embedded. Suppose we have an IC, and on that IC we put all the, we can put thousands and thousands number of different components. So the device are become compact, but giving us more features. So the passive lamp circuit elements like resistors, inductors, and capacitors, those are the basic building blocks of an electronic circuit. And that was thought for over 150 years. But in 1971, Leon Osua has put forwarded that there is another passive element which is known as memristor. Memristor means memory resistor. And just like resistor is related with voltage and current, capacitor is related with charge and voltage, inductor is related with current and magnetic flux. In that same way, that one is related with flux and voltage. And for this flux and memory resistance means the resistance is not linear, it is non-linear. Non-linear means it's varying with time. So in earlier cases, we thought that just like Ohm's law, equal R is the static one. But when we are speaking about memristor, it is the dynamic one. With time, with space, the resistance changes. That's why there is the researchers and scientists are thinking that this memristor may replace our human brains also, because it can store, it can, there is the possibilities of on and off of our memories. So that may be. So we'll not speak on this lecture about the memory. So we will focus on another two devices. So this resistor, capacitor, and inductors, these are the main passive elements which are used for any device design. So now in nanoparticles, how this will behave, because for a nanoparticles or a nano device, or suppose like micro size device, we can put big elements. So what we have to do, we have to develop some new architecture, and for that development of new architecture, we have to scale down, and we have to think of the nanomaterials, whether these nanoparticles will give what type of behavior this nanoparticle shows. So just like when our permittivity, real part of the complex effective permittivity is greater than 0, then it is found that it will show the capacitive behavior. That means those nanomaterials which has effective complex permittivity, real part is greater than 0, can be used as a capacitor. And those material which is having real part of the complex effective permittivity negative, that can be used as inductor. And where imaginary part is not 0. For those materials which is having both real part and imaginary part in that dielectric profile, that can be used as a resistor. So when we consider, for suppose silver, gold, for those real part is negative. And if we think of some semiconducting materials like cadmium sulfide, zinc sulfide, etc., and many more are there, zinc oxide, etc., those shows the capacitive behavior. And for the imaginary part, so if we synthesize in that way, then for cadmium sulfide also imaginary part will not be 0, and for gold also imaginary part is not be 0. So in that way, we can make the circuits. Apart from spherical nanospheres are utilized most widely, and apart from nanospheres also other nanostructures such as rectangular, triangular, etc. can also be paired up to act as series and parallel nanostructures or nanocircuits. If we take two nanomaterials, suppose one which is having its permittivity, real part of the permittivity less than 0, and one with permittivity greater than 0, then it will give us an LC circuit. So in that way, we can design different types of circuits for different series or parallel connections using nanostructures such as rectangular, triangular, etc. And as we all know, this is the core cell nanostructure. In the core cell nanostructure, it is a heterogeneous structure. So here core is there, and the core layer, on that core a layer is there which is known as cell. And when we are speaking about a matrix-based nanostructure, there will be an host matrix, and that host matrix, here suppose I have given air, and if this air is replaced by suppose a polymer host matrix, suppose it may be a polyvinyl alcohol, stars, or many other things. So there these core cell particles are embedded. So this will make a nanostructure, and this can be used, or this may act as a nano device. So there are different types of core cell structure. It may be metal core and different metal cell, metal core and dielectric cell, metal core and polymer cell, semiconductor core and semiconductor cell, metal core and semiconductor cell, polymer core and polymer cell. And in this case, polymer core and polymer cell, the core is of different types of polymeric material and cell is of different types of polymeric material. And in this case, dielectric core and dielectric cell, polymer core and dielectric cell. So according to the goal of the use, we can use, and people are using different type of constitutive materials which can be applied. So this polymer matrix core cell structures has wide applications, including organic light emitting diodes, organic photovoltaics, sensors, as well as biomedical and pharmaceutical applications due to their advantages, because it is low cost, it is easy, it has, with the polymeric matrix, we can fabricate the device easily, and it is transparent also, and it has, due to the transparent property also, it becomes more fascinating, and its application is increasing day by day in the optical domain. So this core cell nanoparticles has wide applications in the biomedical field, like bioimaging, drug delivery, biosensing, then detecting cancer cells, then therapeutic applications, and many more. Already we have attended the lectures during these 4 days in very details. So now I am going to the device. The first device is an antenna. So when we are using the targeted drug delivery, in the drug delivery system, we have to deliver the drug. So to deliver the drug, we need a carrier. So these nanoparticles or nanostructures can work as the drug carrier. So these drug carriers, we can make it directive one, and then it will behave as an antenna, just like the normal antenna. When that antenna which we are using, antenna is using for transmitting and receiving signals. So for this drug delivery systems also, we need a system, the system which will receive and transmit the drugs to the tissues or cells. So for that, we can use the single component nanoparticles or double component core cell nanoparticles. For this nano antenna, basically dielectric and metal are used. And these metallic nanoparticles are known as plasmonic nanoparticles also. And these plasmonic nanoparticles has the special properties of surface plasmon resonance. And for that, we can use that in optical domain. And in optical domain, just like in radio frequency, voltage and current will govern the system. In that way, in microwave frequency, transmission and reflection will govern the system. In that way, when we speak about the optical domain, the properties will be the scattering, absorption, and emission. So this electric and magnetic field will be there when in this system. This one is the scatterer. This scatterer is the maybe a single component or a heterostructure double component, and this one is the matrix. This matrix is polymer, and in this case, this matrix is used. The matrix used is polyvinyl alcohol, and here this is the perfectly matched layer. A layer is there because otherwise the system will be infinite. If from the application point of view, a system cannot be infinite, it will have some boundary. So this is the boundary. There will be a layer. This layer is this one is the air. So these are the symmetry in the electric field, and there will be symmetry in the magnetic field, and there will be an incident plane wave. And for the scattered field formulation, this electric field, then the wave number and the propagation of the signals all will work simultaneously. And if a plane wave is incident on an object, then the amplitude of the scattering object will depend at its distance. So it cannot be infinite. It will be finite. And for finiteness of the structure, perfectly matched layer is there. And now how we can scale down this behavior? For the bark metal particles, as we know, Lorentz-Drude model is there. And in that Lorentz-Drude model, we have found that this is associated, this is here, bound electron plasma frequency is there, bound electron decay time is there, incident radiation is there, and scattering time is there. Means for the bark metal particles, the permittivity is governed mainly by the plasma frequency, decay, and the free electron scattering. But here there is no term or associated with the size. But for nanoparticle, since size is the most important parameter, and by varying the size for the same material, we can found different properties for and we can utilize it for different application. Just like if we have, suppose we have a semiconducting material whose size is comparable to the de Broglie wavelength, means if it is a quantum dot, its size is within 10 nanometer, then we can use it for different application like solar cell or this type of application. But when we have a suppose gold nanoparticle also, if its size is smaller, its application is different. So, and for obtain the scattering behavior, suppose with that there will be a particular limit. We cannot take much smaller nanoparticles or very much bigger nanoparticle. So there is a particular band of size. So for metal nanoparticles of size smaller than the mean free part, the decay time has been proved to be size dependent. And if it is size dependent, the size dependency can be realized by gamma f. This gamma f factor is equal to 1 by T naught plus 2 Vf by d. Here Vf is the Fermi velocity and d is the diameter of the particle. Means this is the parameter where size dependency is incorporated. And if we incorporate the size dependency, then the equation is modified in this way. Here there is a factor which is associated with the size. So for varying size, we can have different permittivity profile. And this polymer matrix, which has very well transparent nature, so this polymer matrix and homogeneity is also achieved for polymer matrix, and basically for polyvinyl alcohol, for this type of optical devices, the homogenization and dispersion is very promising. So earlier one, this is for the particle. Now this is a structure. In this structure, we have this two-layer core cell structure is there and a matrix is there. So only this equation cannot give us the information of this system. So to obtain the information of this core cell nanoparticles, again there are without any other reason, if we consider that this is the core and this is the cell, and if we think that there is no gap between this core and cell, then we can, there is the size dependent permittivity, and along with size it is frequency dependent also. And for the whole system, for this whole effective medium, this is known as the effective medium. Since here a matrix is there and the nanoparticles, here I have shown one, but here number of nanoparticles, just like I had shown earlier, number of nanoparticles are there. These nanoparticles along with this polymer matrix makes it an embedded system. And if this is the embedded system, then for the whole system, volume fraction comes into play. Means the size as well as the size ratio of this core and cell material and the volume fraction, means at what concentration of this host matrix, what is the weight percentage of the nanomaterials, that will also be the main important point to achieve the goal as for different required applications. So these are the real and imaginary part for different core cell size ratio. Here for this, the core is silica and the cell is gold. So we have found that there is a core cell size ratio. This ratio also, this core and size also, we cannot put arbitrarily. We have to engineer the size of the core cell size ratio to achieve the effective dielectric properties. In this is the real part, and this real part is negative, and this real part is negative. That's why this plasmonic or surface plasmon resonance comes into play, and this is the main reason this permittivity profile of the dielectric metal or plasmonic nanostructure, which make it useful for optical device design. And this is the imaginary part, as we have said earlier. This is the negative part, so it can be behave as L, and this imaginary part of permittivity is positive. So this positive part, this may be resistance. So it will, if we make a circuit, then this L and R will comes into play. And this, that domain, the region where the wavelength is 620 nanometer. And for different size parameters, if in these studies, the size is kept fixed as 71 nanometer. And for that 71 nanometer, here the total size is same, 71 nanometer for all the cases. And the size of the core and size of the cell are varied. So by varying the core and cell size ratio, we have found that the 64/7 is redshifted. So this can be moved towards the higher domains, near infrared domains also. And in case of imaginary part here, and another important point is that in case of real part of permittivity, up till around 500, they are almost showing the same behavior, and after that it is varied. And in case of imaginary part, we have found that up till around 500, it is varying, and after that it becomes static almost. And if we compare it, the imaginary part of 66/5 ratio and 64/7 ratio and 66/5, these are the real part and these are the effective parameter. So what we have found that the best we have achieved for 64/7. So this size ratio for silica gold can be used for optical device design. So as already I have said, in the optical domain, scattering is the main parameters. So we all know that just like the blues color of light that is given by Rayleigh scattering, there is another scattering, that one is known as Mie scattering, M-I-E, Mie. The scientist has invented that, and this Mie scattering is associated with the electromagnetic light wave, and this, as light wave is electromagnetic in nature, so this Mie solved that with the help of the Maxwell's equation. As we all know, there are the Maxwell's equation is the main equation or main field theory for which our communication system is there, because the Maxwell's equation, in the Maxwell equation, there are 4 equations, as we know, and there is a time dependent part is also there. So in the time dependent, so that is for a dynamic system. And so this Mie solved the Maxwell's equation for an electromagnetic light wave interacting with small metallic spheres. And for this my scattering coefficients, I will not go so much detail of this. So what we have found that the resonant condition for core cell nanoparticles is satisfied for dielectric material, and the polarizability is maximum. That means scattering coefficient is maximum. And in case of a homogeneous scatterer, the presence of scattering related to the surface plasmon polaritons on the surface of the scatterer. So that's why it is important from this my scattering concept. It is important that the cell material should be a plasmonic material. And by properly tuning the core cell size ratio, the resonance condition can be achieved. And this plasmonic nanoparticle and core cell nanoparticle, where the core is dielectric and cell is metallic, that results in a plasmon derived optical resonance, and which make it for efficient coupling of electromagnetic radiation to nano devices, and which opens up broad range of applications. So these are the surface plasmon resonance wavelength graph, where for varying different sizes, the surface plasmon resonance wavelength are calculated, and here it is found that the surface plasmon resonance peak is dominated for the smaller size particle, which is around 10 nanometer to 30 nanometer, and for the larger size particles, which is for 40 nanometer to 120 nanometer, it is dominated by scattering. So it can be said that for this small size particle, since the surface plasmon resonance peak is dominated by absorption, so it may be used as absorber. And since for this size band 40 nanometer above, this can be governed by the, since it is dominated by the scattering, so it can be used for antenna application. So these are the extinction profile. This is the extinction efficiency versus wavelength for gold and silver. So where we are getting the resonating at 400 nanometer and at 617 nanometer. So if we compare gold and silver, then what we have found that this gold nanoparticle is found to be maximum at 600 nanometer. That means it is redshifted, and this for a silver it is at 400 nanometer. So if we want to use it for medical applications, then compared to silver, gold is more promising and more suitable for targeted drug delivery application. So this, along with the size, there is another parameter which is directivity. Directive means how much directive, how directly or acquiring small area, how directed drugs can be put. So these are the dipole source, and as we know, when we are targeting the drug, then if we are using these nanoparticles as the carrier, then this gold nanoparticle will work as the dipole source element, and this gold, which is this is not for gold and nanoparticle of 70 nanometer, this shows far field radiation pattern, and this one is 40 for 40 nanometer. It is a little more directive, but dipole source we are not achieved. So for gold nanoparticle, when core size is 66 nanometer, the resonance wavelength is 594. For 60 nanometer, 590. For 50 nanometer, we are getting the resonance wavelength 570 nanometer. And for 30 nanometer particle, we are getting the resonance wavelength at 520 nanometer. So from this study also, when PVA matrix is the host medium, then what we have found that if we are taking gold nanoparticle, that means single component nanomaterial, then for 66 nanometer, it shows the promising result. And in case of silica core and gold cell, when this core size is 66 nanometer and cell is 5 nanometer, then the resonance wavelength we are getting 990 nanometer. That means when we are using a single component, then what we have found that the resonance wavelength is 594 nanometer. But when we are use the layer, gold layer as a cell material, then what we have found that for this we are getting the resonance wavelength 990 nanometer. So it is towards the NIR region. So when we are using gold core and silica cell, so this is the different study. Here only gold nanoparticle is there, and for this gold single component gold nanoparticle, when the size is 66 nanometer, resonance wavelength we are getting 592 nanometer. And when core is silica and cell is gold, we are getting for only for 5 nanometer cell size. For this 66 nanometer core, we are getting the resonance wavelength at 990 nanometer. When the core and cell material is changes, when gold is used as core and silica as cell, then we are getting that it is reducing. It's around 730 nanometer. And for silver core and gold cell, it is 690 nanometer. And for gold core silver cell, it is 730 nanometer. And for silica core, and for silica core and silver, so we are getting is 520 nanometer. So from this study also we have got that for gold nanoparticles, variation in resonant wavelength is 74 nanometer. Here 594 nanometer, and for 30 nanometer size particle it is 520 nanometer. But for the core cell nanoparticles, when thickness variation of only 6 nanometer, keeping total size fixed at 1 nanometer, cause the variation of resonance wavelength of around 550 to 30 nanometer. So the size is fixed at 71 nanometer total size. And by tuning the core and cell size ratio, what we have found that the resonance wavelength is varying from 50 to 200 nanometer. So this comparison also shows that core cell nanoparticles is much more suitable, and in these core cell nanoparticles, the cell materials properties or the layer of the cell material which make it very thickness of this layer is very sensitive to the resonance wavelength. That means if we can tailor the cell material and cell size properly, then we can use it for different application. And from this table we have found that for silica core and gold cell, when we are using core of 66 nanometer and cell of 5 nanometer, then the resonance wavelength is 990 nanometer. So from this study also we have found the silica gold core cell nanoparticles of core size 66 nanometer and cell thickness 5 nanometer, resonance wavelength is found to be highest. In 2-3 earlier lectures also, people are focused on the silica gold in different point of parameter. And from this electrical properties also, it is observed that the silica gold core cell nanoparticles, when we tuned the core and cell size properly, and if we choose the host matrix properly, then the resonance wavelengths can be redshifted to the NIR region. So by varying the cell thickness, if we vary the cell, suppose 3 nanometer, 5 nanometer, and 12 nanometer, then also we have found that this 5 nanometer shows the promising behavior. That means there is a particular size band and there is a particular size of the thickness. If we make the cell more smaller in the point of view of the thickness, or if we make it more larger, then also it is not that higher the cell thickness, higher will be the resonance wavelength. It is not like that. So by proper tuning the size of the cell material, we can achieve resonance wavelength redshifted. And if we take the core size as 66 nanometer, and if we use different cell thickness, then also we have found that at 5 nanometer it shows the maximum resonance wavelength. So materials with resonance wavelength in the IR and NIR region are of great technological importance from the applications, just like medical imaging, treating tumors, sensing, microscopy, spectroscopy, and different remote monitoring of industrial equipments. And this graph shows different type of core cell structures and the extinction efficiency, where the core is 66 nanometer and the cell thickness is of 5 nanometer. And from this study also we have found that the silica gold falls in the NIR domain. That means if we compare the extinction efficiency also of this material, then the silica gold is redshifted. So that is more suitable in comparison to the other materials, though here in all the 5 studies, plasmonic materials are associated, but this silica gold shows the promising behavior. So this is the varying core to cell size ratio, keeping total size fixed at 71 nanometer. And here also what we have found that if we take the core as suppose 71 nanometer is fixed. So if we think that if we make the core size smaller and cell size bigger, then what will be the extinction? What is the extinction efficiency, or what is the adjustment of this cell and core? Then also what we have found that 60 by 11 means core size is 60, cell size is 11 nanometer, and in one case core size is 50 nanometer and cell size is 21 nanometer. In this case, the core size is smaller, it's 30 nanometer, and cell size is 41 nanometer. When we have done this comparative study, it is found that for 66 nanometer core size, and when thickness of the cell is 5 nanometer, it shows the better result. So lambda naught is found to get redshifted for silica gold core cell nanoparticle, and if we compare with single component gold nanoparticle, then it's by around 500 nanometer. So earlier people were using the single gold nanoparticle as the source. So after getting these encouraging results, and many other research groups has also experimentally doing that, and then now it is observed that these silica gold core cell nanoparticles are used as the dipole source for targeted drug delivery applications. So earlier it is observed that before 10 years or 7-8 years that silver is basically used for drug functionalization in human body. So but nowadays it is found that core cell structures are used, and one potential candidate for that is silica gold core cell nanoparticles. Now, while most of these research works related to the drug delivery and why it is used polymer matrix, people can use other material also, but here a little comparative study is there. Here polyvinyl alcohol, water and starch is taking. So what we have found that when we are using the polyvinyl alcohol as a matrix, the redshift is more. So for that reason, proper selection of host medium is important to enhance the efficiency of a nano device. First, people choose the material. Which material will serve the purpose? So first, people choose the nanomaterials. People study the behavior of the nanomaterials, chemical behavior, physical behavior, then electronics behavior, biological behavior, and to accommodate these nanoparticles, we need a matrix. And proper selection of that host matrix is very important for efficiency of a nano device. So this is the polarizability study. And for this polarizability study also, when the core cell size ratio is used to determine the size of the reflector and directors. So before that, this one is the typical design of an Yagi-Uda antenna. As we all knew that antenna is used for communication, just like in our television, earlier an antenna was there. So in that antenna, there is a feed element is there, and there are 4 or 5 rods are there at everyone's home. From road itself, people able to knew that this particular house is having a TV like that. So that antenna, for that also that antenna works as a transmitter and a receiver. And in case of drug delivery system, these nanoparticles are used as transmitter and receiver. And these nanoparticles, when we are arranging in a proper way, and if we can tailor the size ratio appropriately, properly, then it can behave as an antenna. And this Yagi-Uda antenna is the most brilliant and realistic idea from the point of view of targeted drug delivery application. Though this area of research is started from 2007, but within these 13-14 years, tremendous development in this research area has grown up. And here this is the feed element. That means this is the active element. And these are the reflectors and directors. So these are the passive elements. So here, since we are speaking in the wavelength nanometer point of view, just like here when we are speaking about radio frequency, we speak in meter. But when we are speaking about the nanometer, so in terms of wavelength, we are speaking that these are 0.25 lambda. That means suppose our lambda is 600 nanometer. That means if this lambda is 600 nanometer, then the distance between the feed and reflector should be 0.25 into 600 nanometer, that amount. And these are the directors. And from study also, it has found that here, higher the number of reflectors, the properties may change. But if we increase reflectors numbers, it doesn't show significant results. So in most of the cases, that's why there is one reflector, and these directors may be 3 directors, may be 5 directors, may be 7. But to make it more compact, people are studying, and for designing an ultra compact device, people used smaller number of directors also, just like the reflector. So this is the antenna structure, just like this Yagi-Uda antenna. Here, this one is the feed element, and these are the director, and this is the reflector. Just like the TV antenna, in the TV antenna there are some rods. So these rods, if we think that these rods are replaced by some core cell nanoparticles, then the structure will be like this. So here, this one is in case of the antenna, Yagi-Uda antenna of that radio frequency. In that, these are the rod of some centimeter. So in this case, these are some nanostructures. Here, these are silica gold nano core cell nanostructure or nanoparticle is there, and the size is nanometer, obviously. So the design, this is the size is miniaturized. So this is the feed element, and this is the reflector, and this is the director. The another important point of this diagram is that here the material is same. The same material is used to design this reflector, director, and feed element. Just the size ratio is tailored. By tailoring this or tuning this core to cell size ratio, the same material is using as feed element, reflector, and director. And the incident plane wave is propagating in the x direction, and the electric field is polarized along the z direction. And here, this feed element is at origin. That means x equals 0, and this reflector is at x less than 0. That means the direction of propagation is positive direction in the x direction, and the feed element is placed at the origin, and the director is at the positive direction, means along the direction of propagation, and the reflector is at the negative direction. And this DSR and DSD. DSD is the distance between the source and the director. As already we have discussed that there is a particular optimized distance. So if we think that we put this feed element here, and at any distance we put the reflector or directors. But to obtain optimized result, we have to put it by obeying the basic theory of antenna, classical theory of antenna. So this is the feed element, this is the director, and this is the distance. So the antenna elements of our different size ratio, and these and nanostructures are in the host matrix, polyvinyl alcohol. So this is an effective medium, and in this effective medium, these are obviously air, because we are making the host matrix is our polyvinyl alcohol. There we are putting this silica gold nano core cell nanostructures, which are having different size ratio at different distances, and air is, and it is surrounded by air, so it is a unit cell. So this whole system we can consider as a unit cell, and these boundaries, the boundaries are completely transparent and non-reflecting for the radiated wave. And the length and height of the unit cell is, the length is 1368 nanometer, and the height is 613 nanometer. That means if we use the Yagi-Uda antenna structure, and in that Yagi-Uda antenna structure, if the feed element, reflector, and directors are replaced by nanostructures, and as we have said that in comparison to the single components just like gold, silver nanoparticles, since the extinction efficiency, rate shifting in all the parameters, the silica gold core cell structure is suitable one. So this feed element, reflector, and director are made by silica gold nanoparticles, and the sizes of differences. And here also another important point is there. In this case, here also this core total size is 71 nanometer. That means this whole size is 71 nanometer, and the core and cell size we are varying to get the reflector, director, and feed element. So in this case, in this comparison of extinction efficiency, where embedded in polyvinyl alcohol matrix, single element for this single element nano antenna, and this is compared for the validation purpose with the Oldenburg et al. Model. And what we have found that when the core is smaller and cell is larger, the lambda naught is around 750 nanometer, and which is in the feasible range. But when the core size is increases from 63 nanometer to 68 nanometer, with decrease in cell thickness from 8 nanometer to 3 nanometer, 800 to 1050 nanometer range of resonance wavelength is achieved, and which is in the NIR wavelength range. So from these also we can say that when the core size is outside the size band, it gets blue shifted. That means it doesn't necessarily mean that if we increase the core size or if we increase the cell size, then red shift will be there. There is a particular size band, and for silica gold, the particular size is 71 nanometer, and for that particular size band, if we tuned it properly, then we can get optimized result. And from the directivity point of view also, for 66 core size and 5 nanometer cell size, the resonant wavelength at 990 nanometer and the directivity is 9.8996 decibel and the gain is 16.033 decibel. In comparison to the other core cell size ratio, this shows more promising behavior. And these are the some radiation pattern, because we need directive one. So for targeted drug delivery, so we can't use some type of carriers which radiates in different directions, because it can make harm to other healthy tissues also. So for that, utmost care must have to be taken when delivering the drugs. So here also single silica gold core cell nanoparticle as a single element antenna offers much higher directivity and gain compared to many other optical nano antenna of various types. So we have various types of nano antenna, but this Yagi-Uda nano antenna is the most optimistic type of antenna we can say for targeted drug delivery applications. The another important point, as I have said earlier, that this size matters. The distance and the size matters. So this distance between the feed and reflector, the distance between the feed and directors should be optimized. And in this silica gold case, along with the reflector and director size, the distance also plays a very important role in the design consideration. And for this design consideration, what we have found that when the source and for suppose 990 nanometer, for 990 nanometer lambda, if we follow this distribution, then we have seen that the director's distance between the feed and director is larger or more in comparison to the distance between the feed and reflector. So obeying this basic theory of classical antenna, here also these are the source and reflector distance, and this source and reflector distance also we are getting a window, and this optimized distance between the feed and reflector is 160 nanometer. That means 0.16 lambda naught, and here the directivity is achieved as 16.28, and the optimized distance between the source or feed elements and director is 240 nanometer. That means it is 0.24 lambda naught, and here the directivity is 16.32. That means the basic classical design consideration for reflector and director is applicable here. So when this has been done, then it is found that, suppose another important point is that the number of directors may be increased. So someone may think of, why one director only? In the classical YAG, the typical Yagi-Uda antenna, there are more numbers of directors. So when we increase the number of directors also, it is found that with one reflector and 4 directors, the optimized result is achieved when we are taking the silica gold, only silica gold core cell nanoparticle. That means if we consider the single core cell nanostructure or nanoparticle as an structure for the design consideration, then what we have found that the optimized result is achieved for 1 reflector and 4 directors. But in this case, when we are increasing the number of directors for this Yagi-Uda and designed Yagi-Uda antenna, then what we have found that this directivity, this optimized directivity is achieved. Here no radiation is there in any directions. That means this will target the particular area. Means when we will use the silica gold core cell nanostructure with 1 reflector and 1 director, then we achieve the optimized directivity for, and that means optimized delivery of the drugs. So the maximum directivity and gain is achieved are 16.32 and the gain is 24.28 decibel. So super directive radiation pattern is achieved for 1 reflector and 1 director. That means if we use polymer matrix based core cell nanostructures, polymer matrix based core cell nanostructures means where our core is dielectric and cell is plasmonic. If we used in that way, and if our host matrix is polyvinyl alcohol, people can try for other polymeric matrix also. So then what we have got, that ultra compact and directive targeted drug delivery can be achieved. So from this study, it can be concluded that with silica as core and gold as cell material, and this silica gold core cell nanoparticles as feed element, reflector and director, we can achieve optimized drug delivery systems at the NIR region. And since this is ultra compact, so definitely size will be miniaturized and directivity is also very promising. So it can be used for targeted drug delivery application. So now moving to the second part of this talk, there is a design of a filter. We know that basically filter is the simple concept. In case of our, suppose, water filter also, we filter the water so that we can get pure water, means without any impurities. So in that same way, when we are propagating the signals also, according to the applications, we want different types of filtering signals for different medical applications also. So here this technique is microwave photonics techniques, since this will work at optical region at very higher frequency of microwave band up till to 20 gigahertz. So here photonics techniques has been used. Earlier photonic techniques has been used for the last 20 years or more than 25 years for telecommunication instrumentation, then antenna systems, radar systems, etc. And for cellular technology, wireless communication technology. So now the researchers are thinking of using these microwave photonic techniques for biomedical imaging with application of nanostructures. So modification of fundamental architectures by scaling down the component size using nanoparticle will have great influence on future commercial communication and computing systems as well as in medical technology. And this microwave imaging is a technique which is used to observe the internal structure of an object, just like the computer tomography, CT scan. Then in CT scan also there another CT scan where dye is injected to the body and to get the detail of the internal structures, minute things. In that way, this microwave imaging technique is also a technique which is used to observe the internal structure of an object where electromagnetic fields is there through the different interface of the human body. And this microwave imaging techniques has been revealed to be found to be useful for detection of breast cancer. And here also this dielectric property plays a very important role because the research has said that dielectric property of cancerous tissues are minimum 4 to 5 times larger compared to normal tissues at different microwave frequency bands. And definitely when we go for new techniques and new design, then always new algorithm, new experimental techniques is there. So now when we do the experiment or when we design a filter where nanostructures are associated, then also a question comes into mind whether the techniques which are used for microparticles or which are used for large size devices, will that techniques applicable for this nano also? So the same techniques with some modification or by different types of calibration methods we can use for several diagnosis applications of medical technology such as blood flow pressure monitoring, fluid or water accumulation, brain stroke, etc. has been extensively covered by the spectrum of microwave sensing and imaging. And this extensive application makes it very promising for medical technology. But very small number of studies till now has been reported and going on in the domain of medical technology with an extensive study from the electromagnetic point of view. So the study of dielectric properties and scattering properties of negative nanostructures opens up the possibility to develop new technology for disease detection. So here cadmium sulfide, zinc sulfide are synthesized separately. We will not go for the details of the synthesizing process as many of the earlier speakers has already focused on that area. And cadmium sulfide, zinc sulfide, double component heterostructure core cell has also been synthesized. And here also polyvinyl alcohol is the host matrix. And by varying the molarity and the pH value, different samples are prepared, and by tuning the core and by changing the concentration of core and cell molarity and by varying the pH factor, core cell structures are designed. And these are the XRD patterns, and this XRD pattern also shows that particle size is found in the range of 9.07 to 10 nanometer. We can say quantum dot is synthesized. And these are the some HRTM image. And in this HRTM image, what we have found that the novelty of this synthesis is that here the agglomeration is less, and we are getting the structures the core cell structure in more distinct way, which earlier also we worked in this, and at that time for the cadmium sulfide, zinc sulfide structures, this type of spherical cell coating has not been found. But in this work, this is a very recent publication. It has been published in IEEE TMS, Journal of Electronic Materials, in this January 2021. In this, we have got this spherical structure where coating is also very distinctly spherical. So when we are doing microwave characterization, lot more techniques are available, but since this will be in liquid form, since our nanomaterials are in liquid form, so we cannot use the transmission reflection type of thing. So we are using here open-ended coaxial probe. Open-ended coaxial probe means this is the schematic diagram of the experimental setup. Here the liquid nanosample is there, and there is a vector network analyzer. Here the single port is used, coaxial cable is there, and here this adapter is there, and this probe is touching the surface of the liquid nanosample. And before doing this, calibration has been done, and the calibration of the vector network analyzer was done using 3 different loads: air, short circuit with metal contacts, and distilled water at room temperature. So after doing this, at first the water is measured, and what this is the practical laboratory view of the measuring setup. And here, when it has been found that for water, this is 80. That means that this study earlier, this is the first study for water at high frequency. Earlier at low frequency it is there, and at low frequency, as we all know that water has the permittivity or dielectric constant is around 80, and in this case we have found that at higher frequency it is 500 megahertz. So here also up till we have done the study up till 20 gigahertz, and here we have found that the imaginary part and real part is changing, the permittivity profile is changing in this way. And from biomedical point of view also, this study is very important, as water is one of the main component of our human body, and this permittivity profile of water can be taken as a parameter to determine the electrical behavior of human body. And because when we give drugs, there is a question: when we give drugs or when we deliver drug, when we deliver targeted drug, definitely penetration will be there, the radiation will be there, so the signal will penetrate. So up till what extent it is penetrating, that study has also been done, or that information has also been with the medical practitioners. So this study may reveal that idea of optimal condition for wave penetration, means up till what the drug should be penetrated. So these are the permittivity profile, and here we have observed that this study is up till 20 gigahertz. So this is not a linear one, this is a non-linear one, and here switching is there. We can say it as switching, means the relaxation mechanism is working. So somewhere it is on, somewhere it is off, like that. And this is the imaginary part of the permittivity profile for the cadmium sulfide. And these are the dispersive nanomaterial, and this the permittivity profile of this dispersive nanomaterial is non-linear, and this agrees with the permittivity characteristics of dispersive material. And this is the first reporting of for this cadmium sulfide, and this is the dispersive properties. But for zinc sulfide samples, what we have found that this non-linear or switching pattern is not there. For zinc sulfide, we have found that these are similar to other dielectric materials, and it has small effect for the carrier trappings. But for cadmium sulfide at higher frequencies, it is observed that this behavior is non-linear. And for core cell nanoparticles, what we have found that this behavior is non-linear up till 20 gigahertz. The study has been done from 5 megahertz, 500 megahertz. So it has been found that this is non-linear, and the relaxation mechanism is more stable in comparison to cadmium sulfide. Here cadmium sulfide is the core and zinc sulfide is the cell, and this zinc sulfide shows the dielectric profile just like the other dielectric materials, and this cadmium sulfide shows the permittivity profile which is not linear. And this heterostructure cadmium sulfide zinc sulfide nanocomposites shows larger dielectric constant with increasing trends of trapping rates and detrapping rates. And trapping and detrapping zone is obtained almost in an equal interval time. And this distinguishable trapping and detrapping zone would be controllable by signal frequencies. As we have seen here, the frequencies are different, and at different frequencies we are getting different effective permittivity value, and the peaks are also different. So it can be used for, it is useful for wireless communication also, and it is used for different memory applications also. And this advocates the applicability of this cadmium sulfide zinc sulfide core cell nanostructures for biomedical applications. So these are some earlier graphs in the first series of this talk I have shown. This is for the cadmium sulfide zinc sulfide itself. Here what we have find that in this case these are the cell material. S2 is the cell material, S1 is the core material, and this S3 and S4 are the core cell material. But in this case we have not got this non-linear behavior. So that means when we, if we can control and if we can tune the concentration, size ratio, then environment of the medium, effective medium, we can achieve different properties from the same medium, same material for semiconductor material also. Earlier it was, as we know, I have shown in some first slides also, my first slide also, that for gold nanoparticles for different sizes we are getting different colors. That means the same materials for different sizes we can use for different application. So this study reveals that for semiconductor nanomaterials also, if we can tailor or optimize the concentration, if we can optimize the synthesizing time, then the heating effects, then we can use that material for different applications. So these are the, just like the electric modulus, we know that in all the cases we know how the on and off is going on. So this electric modulus pattern also shows us the clear relaxation peaks and the switching mechanism of the cadmium sulfide zinc sulfide nanostructure at microwave frequency. So this electric modulus also give us the information that this can be used for switching and memory devices. So it can be used for medical applications. So the recorded complex permittivity profile shows that the dielectric profile remains both reversible and stable for all the cycles. And there is that the relaxation and switching behavior shows that for high dielectric state and low dielectric state. That means it's just like the switch. That means it can be used as a switch to behave sometimes as at on state and sometimes at off state. And the switchable materials show promising applications in the field of electrical and electronic devices, for data storage and different other applications. And the switching dielectric behavior achieved in semiconductor core cell nanoparticles. This is the first reporting for that the cadmium sulfide, zinc sulfide nanoparticles can be used as a switchable materials, and this relaxation mechanism can be used to analyze ionic conductivities also. If the ionic conductivity can be used, then this relaxation mechanism can be studied, and this relaxation mechanism or this relaxation profile will give us the information about the charge trapping and detrapping. So this is the modulus, and this frequency dependent complex electric modulus plane plots for cadmium sulfide, zinc sulfide nanosample, and this is in agreement to the Cole-Cole equation. And this also confirms that the behavior, the relaxation behavior is close to Debye relaxation behavior. So by dispersing engineered nano-sized single component as well as double component particle, where polymer is a capping agent, it is possible to achieve filter having all the properties just like our classical electronic filter. And here the advantages are that we are synthesizing the particles, so we can control the concentration, we can control the size, we can control the shape, and with proper choice of the host matrix, we can dynamically control the electrical properties for different applications. And these are the some experimental studies for the, and this is the prototype of the design, and this design is based on the microstrip technique, where electrodes are there. These are the electrodes, and here the core cell nanoparticles embedded in polyvinyl alcohol matrix are there, and this is the substrate. This substrate we can use different, and some may try for indium tin oxide also, or some any other kind of FR4 or any other type of compatible substrate materials. And these are the design parameters for this device. So here, since the nanoparticles are in liquid form, so we are adding 10 micrometer, 10 microliter active as an active material. Here, if we see this nano core cell nanoparticles embedded in polyvinyl alcohol matrix, this is the active material. And here this is the solution. So we have spin coated that 10 microliter samples, and here the conductor films are copper. This electrodes are copper, and the width of the thin film between the microstrip line is 8 to 10 micrometer. And the thickness is around 200 nanometer, and it is always important that to achieve optimized result, the thickness should be comparable to the thickness of the coplanar structure. So this is the equivalent circuit, and here in the same way, just like I have illustrated in some of my slides, that nanoparticles can behave as a capacitor, behave as a resistor. So for cadmium sulfide, zinc sulfide core cell nanostructures, it has been found that it is just behaving like a low pass filter also. Here resistance and capacitance both are present. So these are the gain curve, frequency versus gain curve, and the phase shift. And here these are the transmission loss and reflection loss for the cadmium sulfide, zinc sulfide. Then this D1, this black one is for core, this D2, this one is for cell, and this one is for core cell structure. So it has been found that for this cadmium sulfide, zinc sulfide, the notch is generated, and in this case this is the reflection loss. In the same way for D3 is for core cell structure, and it had the resonant frequency are at 7.916 gigahertz for single component cadmium nanostructures, 7.51 gigahertz for zinc sulfide nanostructures, and 7.2299 gigahertz with minimum reflection loss, whereas minus 19.698 dB, minus 15.313 dB, and minus 14.594 dB. And the transmission loss we have achieved at 9.3 gigahertz, 9.06 gigahertz, and 11.86 gigahertz respectively for cadmium sulfide, zinc sulfide, and cadmium sulfide, zinc sulfide core cell nanostructure. Another important achievements of this work is that here for cadmium sulfide, zinc sulfide core cell nanoparticles embedded in polyvinyl alcohol matrix, the notch is generated at minus 50.14 dB, and which is very challenging to achieve. And here in this case, by proper and effective tuning of the structures, the notch is generated, and the notch is also very distinct, and it is induced by my resonance. As this is at very high frequency, so it is induced by the my resonance theory. So the as design filter has achieved a comparable insertion loss and peak resonance to the state-of-the-art RF notch filter. And this phase shift of the signals, actually this is the phase shifting, and this phase shift for the core cell nanostructures also shows the properties as microwave photonic filter. And from the application point of view also, the observed behavior which we have achieved for complex permittivity profile of cadmium sulfide, zinc sulfide core cell nanostructure, it is very promising. And since switching mechanism is there, so it can be used for various different applications. And there is potential promise, we can say, to extend this behavior for optical imaging technique. This FTIR spectrum for this core cell structure, core cell device also shows that these are compatible or co-attached with biomedical. So at optical wavelength, the nanosample of core cell device shows multiband notch generation. So the main conclusion we can put from this study is that these optical properties are used to obtain hemoglobin concentration volume and absolute oxygen saturation. And with modulation capacity, since the frequency band is very high and it's up till 20 gigahertz, so it may serve the purpose for some specialized specific optical system to extract spectroscopic information for any kind of solid and liquid phantom. That means that tomography is again, so for CT scan also it may be used. So for accurate clinical imaging, for phantom resembling breast tissue broadband extraction process is preferred, and this present study broadband extraction process is used. And in terms of nanostructure, it is giving the information of both amplitude, as we have shown, amplitude is shown, gain is shown, and for the phase shift also. So all the parameters which are required for diagnosing a disease or for imaging, it may serve the purpose. So this is the end of my today's topic. I tried to cover almost all the things, but since time is very limited, so I would like to acknowledge Department of Science and Technology, Government of India, KIT, Kharagpur Institute of Technology, Germany, Indian Institute of Technology, Kanpur, Guwahati University, Assam, Central Institute of Petrochemical Engineering and Technology, Center for Skilling and Technical Support, Guwahati, and Center for Research in Nanoscience and Nanotechnology, Calcutta University, for providing me all the facilities and all the opportunities and supporting me all the way to carry forward all the research work. Thank you very much. The session is now open for interaction.
Yeah, thank you, Dr. Jutika Devi.
Thank you, sir.
It's really very informative in this field, especially your the nano part, their implementation in biomedical as well as imaging. That your second part of your talk, that imaging of biomedical applications. Your research and development work is that you are showing here. We are sharing today with our eminent participants. It's your expertise is really is beneficial for applying in this biomedical sector. Now I request all the participants to kindly interact and to clear the doubts if you have anything, so that Dr. Devi can address directly. Yeah, we received some remarks from Dr. Geeta Singh. So she is giving thank you for your very elaborating topic lecture today. Yeah, other participants, if you have anything, we can, yeah, Dr. Dharmalingam K has shared that. Nice work, ma'am.
Thank you, sir.
Yeah, Dr. Anupama Rajput and Dr. Gaurav Agarwal is also expressing their thanks for this informative session. Yeah, Dr. P. Sanguwali is also expressing thanks for this informative presentation. Yeah, Dr. Ankur Kausik is also expressing thanks for the session. Yeah, so we are receiving a lot of remarks from different participants. Dr. S. Selvam, Dr. P. Bharati, they are also expressing thanks for the very informative and nice presentation. They are writing their expression. Yeah, I think if we do not have right now, if we do not have any other queries, so I think we can conclude this session. The From on behalf of CIPET Guwahati, we are expressing sincere thanks to Dr. Jyotika Devi for sharing your research in this with nanostructure, how it is implemented for biomedical applications. Thank you very much, and I am 100% sure, we are confident that your talk is definitely so, it will definitely pave the way how we can generate other nanoparticle also with the help of the theoretical concept, how we can generate the we fabricate or synthesis different kind of nanostructure, especially for biomedical applications, especially in the sector of imaging application. So thank you very much, Dr. Jyotika Devi, for your very nice and expertise lecture on this field. And I think now the other program will I think the other program about the other programs, because we are also planning uh, exam today. There will be a objective type of exam. I just to make I just to inform all the participants that uh, after successfully if they can appear in the exam only, the certificate will be distributed. And I am very glad to share that the Honorable Vice Chancellor of University of Science and Technology, Meghalaya, has given his consent to be present uh, in the valedictory function. So I would like to request all the participants to please be on time and be present in the valedictory functions too. So now it is over to Dr. Jyotika Devi to, if you have any other informations to share with the participants. Over to Devi.
Thank you very much, sir. Thank you very much to CIPET CSTS Guwahati for supporting all the way to deliver this lecture. Thank you, sir. So we will have a, dear participants, we will have a test at 2:30 p.m. So now lunch break is there. So please join there at 2:30 p.m. after the valedictory function will be there from 3:30 p.m. So now lunch break is there. You can be stay at same line or you can rejoin again. So please join by the link at 2:30 p.m. and link will be shared here in this WebEx platform. And as sir has said, at 3:30, the Vice Chancellor of University of Science and Technology Meghalaya has consented to be present in the valedictory function. So I request all of you to be present at the valedictory function also to make the event successful. Thank you. Good afternoon, everyone. The test link is provided at the chat box. Test duration will be 3 hours. 25 questions are there, each carrying 4 marks. Total is 100 marks. Mukesh, why has raised your hand?
Hello, ma'am. Hello, ma'am. Hello.
Yes. Yes, please, Ajit Kumar.
Hello, ma'am. I didn't get any link. Hello.
Yes, we are giving again.
Google Form link. I didn't get. I checked mail and WhatsApp.
Yes, in this you will get.
Hello.
Ye-
Hello.
yes, sir, actually link is given at the chat box. So we are giving the link again. Okay.
Okay. Okay. Thank you. Thank you. Give the
Yes.
link again. For more.
Yes, please be in the line. Our valedictory function is going to start shortly. Hello. Valedictory. ডিৰেক্টৰ ছাৰক কৰিছে নেকি? মই কৰা নাই, আপুনি কৰক।
Is he still in class now? Person? Person?
Hello. Yeah. Very good afternoon to all. At the end of these 5 days, we are reaching to the final schedule, final session, like a valedictory function. We are very much thankful that today we have among us eminent scientist, educationalist, a motivator in the sector of education sector is none but Professor Gauridat Sharma, the Vice Chancellor of USTM University of Science and Technology, Meghalaya. A very good afternoon, sir.
Yes.
Yes, sir.
Good afternoon.
Welcome to these 5 days AICT sponsored ATOL Faculty Development Program. On behalf of CIPET Guwahati team, I welcome you on this virtual platform. I think the internet speed from professor's side is a little bit slow. So, respected Professor Gauridat Sharma sir, PC USTM, respected Director and Head CIPET CSTS-GUWAHATI, Sanjay Kumar Chaudhary sir, my dear colleague Dr. Jutika Devi and Dr. Rotikanto Das, coordinator of this faculty development program, my dear faculty members from different departments, and respected all the participants from different institute, different colleges, different research and development wings, and universities, professors, lecturers, readers, and research fellow from all over India. I welcome you all to this valedictory function of these 5 days faculty development program. It is a very prestigious faculty development program sponsored by AICTE Otol. So the whole program was for 5 days, and I strongly believe that the different professors from different IITs, central universities, and different universities, I think you got new directions in the field of drug delivery, in the field of medical sectors, how we can best utilize the nanostructures, where the polymer is the precursor for this matrix system. I would like to, in this occasion, I would like to request our director and head, Sri Chaudhary sir, to kindly brief about this valedictory function, as well as to give a summary report to all the participants and to Professor Sharma sir. Sir, over to director sir.
Good afternoon, sir. Thank you, Dr. Harishchandra Deka, sir. And it's my great pleasure to inform you that, sir, that program is sponsored by the AICTE Atal FDP program for the CIPET-GUWAHATI, which is organized from the 7th February to 11th February. And AICTE has given to conduct that program with to minimum at 200 participants in the field of faculty, scientists, engineers, researchers from different reputed universities and technical institutions and organizations, as well as the industries all over the India. And from last 5 days, we have observed and monitor total 768 participants were attended the program, and for our resource persons also, total 16 resource persons from the different universities participate in this polymer FDP program, polymer matrix based nanostructure for targeted drug delivery application. I am very thankful to the Abhijit Bandyopadhyay from the Calcutta University, Dr. Pradeep Peck, Indian Institute of Technology, BHU, Dr. Garima Agrawal, IIT Mandi, Dr. Ranjan Dattak, Lita, Royal Global University, Guwahati, Dr. Yashveer Singh, IIT Ropar, and Professor Pralay Maheti, IIT BHU, Professor Niranjan Karak, Tezpur University, Dr. Nilima Devi, Dono International Institute of Research, and Dr. Vinay Sharma, IIT Jammu. Professor Sunanda Barua, Assam Downtown University, and Dr. Amit Alexander, Naipur, Guwahati. I am also thankful to the Dr. Harikrishna Deka and Dr. Jutika Devi, those who are from the last 5 days involved and coordinate this virtual platform in CIPET Guwahati. And also I am thankful to the participants of 768, those who are very from the last 5 days. Per day, approximately around the 120 to 130. From the 6 hours, spend the time and get the information about the polymer matrix based nanostructure used in the drug delivery system. And I think when I will take the feedback today and also the conducted by the Jutika that test, found that a main objective of Atal Academy is to imparting quality education in the country and to support technical institutions in research, innovation, and entrepreneurship through training in various emerging areas. We think that we have fulfilled or shared that information through this platform and enrich the knowledge to the participant also. I am very thankful to all the faculty members and also the VC of USTM sir, Professor Gauri Datta Sharma sir, those who have given me this time for the organizing this validity function. Thank you very much, sir. It is a pleasure in Guwahati that we are organizing from the CIPET Guwahati that we are organizing that type of program. So thank you very much, sir.
Thank you, sir. Thank you, sir. Thank you for your inspiring speech and also a brief about the 5 days what we did, who are the faculty members, who are the expert lecturers who taught different topics. Now we are extremely happy and privileged that Professor Gauridat Sharma sir, VC USTM, is with us, among us, as a special guest. And I would like to request sir to kindly and to deliver a speech to motivate us, so that in future also we can do such type of program under your guidance. Now over to Sharma sir.
Hello.
Yes, sir.
Yes, sir.
So good afternoon to every one of you. And in this valedictory function of the faculty development program conducted by CIPET CSTS-GUWAHATI, particularly on the topic polymer matrix based nanostructures for targeted drug delivery applications, which was sponsored by AICTE under this ATAL program. And I was just listening to Professor Sanjay Kumar Chaudhary, the director CIPET, that though as per AICTE only 200 participants were required, but 750 participants who have taken part, that indicates that such kind of the faculty development programs are so important to provide the technological backup to our faculty, scientists, so that they can take their researches for new innovative work and also incorporate the technology. Though due to the Corona and Omicron, mostly because virtually the program has been conducted, but having such a experienced and also eminent personalities as a resource persons from IITs and also from our Northeast, from Royal Global, Downtown, and other institutes, also indicates that we have both locally available resources as well as we can get help from the resources from the renowned institutes of the country. As we know that faculty development program is an important part of NEP 2020, that we need to have continuous upgradation of knowledge so that the researches can be modulated and can be planned as per the changing need of the society, not only to enrich the knowledge, but how this knowledge can be applied and how the knowledge can be made relevant to the market. That means the needs of the society. And in this way, we not only have the theoretical knowledge, but the practical aspects are important, so that in future we can develop our own technology after understanding the proper mechanism of action. Here, particularly when we say drug delivery and its interaction, we have seen that nanotechnological aspects, especially using some of the microbes also, has become now the common drug delivery systems, and these are coming through the new technological approach that how target oriented we can have the drug delivery, drug interaction, and also the action of the drug, so that time taken by the body to reach to the target is reduced. Many now technologies are coming up like this, and using even our theoretical aspects of bioinformatics and some of the platforms where we can also locate how the molecule is interacting with particular protein, and which will be the better site even of that protein, can help us to understand the development of that particular molecule. So this polymer, which is also very important because most of those polymers, they interact the nanostructures which are made of this, how they are taken up, and after going to the target, how they are interacting is an important aspect. I am sure that during these 5 days faculty development program, the knowledge of our faculty from different institutes, especially from Northeast, has helped them to also modify their teaching learning process and pass this knowledge to the students, as both the P and UG David as well as to the research scholars. What we have seen generally that in Northeast we have very rich resources, but because of lack of the applied knowledge, we are not able to develop the intellectual property right, what we call. So this kind of the faculty development programs are also helpful, that we think now how we can have the process of changing in delivery, how we can also modulate different nanoparticles in combinations, or totally searching for the new polymer matrix, which will be more desirable and can be used easily. So in this way, not only gaining the knowledge, but also it will have the critical thinking process among the faculty members, and then they can, through their deep learning through this faculty development program, can be helpful in bringing out some of the which they can carry out and develop some technology useful for the drug delivery system. As we know that recently India has emerged as one of the leading sector, not only for producing the technology developed elsewhere, but in India, for the first time, we have developed the technology, particularly for the corona vaccine, where mRNA-based drug, as well as the drug which is also those strains which are not causing any disease or avirulent, what we call, how they have been taken up and are used to control the targeted organism. And therefore, for the first time, our world recognition, especially from the WHO and other developed countries, has come out that the scientists, they are able to create both the new technology, the technology delivery, as well as they can help the wider spread of such pandemic diseases. Otherwise, we can just imagine what would have happened if this knowledge was not with us. Totally dependence on other countries, not only in terms of cost, but also getting the material. So similarly, the aim of this Atal program under AICT is, because we also have our faculty who have attended this different Atal program under CIDC, that how to apply your knowledge which will be helpful in creating the new application and understanding the mechanism so that we can also enter in this competition of enterprising or creating new knowledge or new business or new solution to the problems faced by the society. This also gives us an opportunity now that under new education policy 2020, we can now create short-term courses, which we call value-added courses on these aspects, and create the human resource which are locally available. And now the NEP 2020 allows us not only to simultaneously go for online courses, even to have dual degree or any other value-added courses, or in replacing some of the courses which are the part of our syllabus. So these program of faculty development to the teachers, they help in thinking in all those directions, that how to now extend, because these 5 days or 7 days programs are to give an orientation, exposure, and awareness, and to sensitize them to the new technology, which may not be possible to give total process of, but experienced people that how they are running these facilities, and this facility can be used there in their institute to have the collaborative work, as well as to come out with some advanced label course. So I am sure that this 5 days faculty development program must have helped a lot, and the mode of the delivery, which was virtual, is during this pandemic also. So I congratulate the Dr. Deka and Professor Sanjay Kumar Chaudhary Sahab, Director CIPET, for organizing such an important online faculty development program. And in future, we would like to join for collaborative such program in number of other areas where we have expertise, so that particularly on the molecular aspects of the trainings, and we can now also provide some physical part of the practical knowledge. so that it brings more confidence and also it helps to utilize those instrumentation which are available here in our Northeast. With this, once again, thank you very much that you have given me this opportunity to share some of my views. Thank you very much and namaskar.
Thank you very much, sir, for your such an inspiring words and path showing lecture topics today. Sir, we strongly believe that under your strong guidance, definitely we can work together in different areas and we can bring the scientific technology. We can utilize this technology for mankind. So we expect that in future also you will be giving the same cooperation, same guidance, so that we can move forward with different aspects of technology and science. Thank you very much, sir. On behalf of CIPET Guwahati team, we sincerely offer our sincere thanks for your time, for your such an inspiring talk. Thank you very much, sir. Yeah, and moving forward to our next agenda, now I'd like to request all our esteemed participants, professors, and research members and faculty members from different institute, organization, and universities. I would like to request you all to kindly share your feedback, kindly share your kindly share your this 5 days journey, how you feel about this faculty development program. So so that your feedback will really motivate us, show us the way so that we can improve ourselves for conducting such type of program in futures. So I'd like to request one or two members, participants.
Sir, good evening, Professor.
Good evening.
Sir, this is Krubagaran, Assistant Professor, Department of Mechanical Engineering from Sri Venkateswara College of Engineering, Tamil Nadu, sir.
Yes, sir.
Yes. So here, 5 days, more than 14 sessions we watched, sir. Each and every sessions, like as a many number of researchers they discussed. So many number of PhD viva, sir. So I am a PhD part-time scholar. So I am doing research in polymer. So lot of things I taken from this FDP, sir. So I thank the coordinators also from the question online MCQ. So what are the different perspective the DC members asking question? I understand the perspective view also from this test, sir. Thank you coordinators and thank you Atal, sir. Thank you, sir.
Thank you, sir. Thanks for your nice comments. And we are also in a process of making the proceedings on these faculty development programs. So I request everyone, if you are also having different web PPTs related to this topic, related to this in line with this our program team. So we'll be publishing these all PPTs lecture series as a proceedings. So, and I'd also like to request all the invited speakers to send their different PPT as well as different their topic related articles in line with this faculty development program, so that each same thing can be circulated to all the participants for our better utilization, as well as we can gather the informations. And I am also privileged, and I'm taking this platform to share that we are also going to organize a 5 days program on plastic product manufacturing and recycling techniques. We will be starting from 15 February to 20 February, and the AICT platform you can join through the same AICT platform, and these whole courses will be conducting starting from choosing of different raw material, their characteristics, what are the different manufacturing techniques are available, how we design a new product, and especially the burning topic, the buzzword like the recycling, pollution of plastics will also be covered in this 5 days series, which will be organizing from 15 February. And you may be knowing like that the new plastic waste management rule 2021, which is an amendment takes place in 2021, how it will be implemented for plastic waste management rules. That thing will also be covered in this short-term training program, which will be conducting from 15 February to 20 February 2022. Just I want to add one news that in different states, including in Assam also, today the Honorable Chief Minister of Assam, Sri Himanta Biswa Sarma sir, has given a news report that in Assam also the single-use plastic will be stopped from the end of say in 2022. So how to cope up with this situation, how we'll move forward, what are the aspects to be discussed, so everything will be included in this training program. So may I request at least another one participants so that they can share their experience for this 5 days training program. May I request Dr. K. Rajendran? Dr. K. Rajendran, sir? Yeah, yeah. Dr. Kalpana Nagpal?
Sir, may I speak? This is Dr. Dharmalingam
Yeah,
from
please.
CBA. Yeah, it is. First, I would like to thank Atal FTP for providing this wonderful opportunity, and CIPET Guwahati, our organizers. Really, it was very wonderful sessions. All the sessions, almost more than 14 sessions, were informative, were very useful, and moreover, I like this. I like the Guwahati place because I have completed my PhD in IIT Guwahati. When the moment I have seen enough Atal FTP and Guwahati place, just I simply, and also my topic also polymer based drug delivery systems. Everything was so nice. So topic and the place. So I just clicked. I was so happy, and to see the northeastern people India. So I love them. So thank you so much for for providing this wonderful opportunity, and looking forward and to come physically and to see all of you in near future. Thank you so much.
Thank you very much, sir. Thank you very much for your very nice comment, and we are very much glad that you did PhD from IIT Guwahati, which is only 4-kilometer distance from CIPET Guwahati. And anytime you are most welcome, sir. We can have different collaborative research work and different kind of the facilities that we have in CIPET, you can utilize. You are always most welcome. AICT is also having the idea so that from different parts of India, the researchers, the professors, we can have a collaborative work in so that we can share our knowledges. We can make combined research and work developmental projects. This is one of the aim, and we believe that we have meet the purpose of this AICT, and from the CIPET CSTS Guwahati team, we really appreciated that all participants is such an highly interactive participants. We hardly get such type of participants for conducting this type of programs. So we are very much thankful to you. Without your strong participations, this program could not have get a success. Thank you very much, sir. Thank you very much to all. So before we move forward to the last session of this program, sir, I would like to request all the participants to kindly switch on your video so that we can have a group photo. Sir, I repeat, all that can we switch on your video so that we can have a group photo. Yeah, I repeat, kindly switch on your video so that we can have a group photo. Sir, I repeat, sir, and all the respected participants, I repeat, kindly switch on the video so that we can have a group photo.
Oh.
Screen. Yeah, thank you very much. Thank you very much to all. Now I'd like to request the coordinator of this faculty development program, Dr. Jutika Devi, to propose his vote of thanks.
Yes. Thank you, sir. At first, I would like to thank AICT Training and Learning Academy Otol for sponsoring this advanced level program on the emerging area related to polymer material, since we are the only institution in India exclusively working on polymer materials. So we are happy that AICT Otol has provided us this highly competitive faculty development program. We would like to thank Eastern Division Regional Office of AICT for helping us all the way by providing different guidance for successful organization of this program. I would like to thank respected Director and Head Sri Sanjay Kumar Chaudhary sir for giving us the mental strength, support and encouragement for smooth conduction of this program. I would like to thank Dr. Hare Krishna Deka, Head of the Department of Training, who has guided and for smooth conduction of the program every day, suggesting and guiding us and helping us all the way. I would like to thank Rotikanta Das sir, PTC coordinator of CIPET CSTS-GUWAHATI, for helping us all the way, starting from preparation, management, and everything for the smooth conduction of the program. I would like to thank our members from the training department for cooperation in all the works related to this program for smooth completion of this program. I would like to thank Utpal Roy, who supported technically without any interruption. For the 5 days, we had able to complete the program. And I would like to thank the participants for their strong participation from day 1, 9:30 to the last event, for making this event a grand success. I would like to thank the resource persons without whom this successful event cannot be a success. Professor Abhijit Bandopadhyay sir from Calcutta University, Dr. Pradeep Paik from IIT BHU, Dr. Gorima Agarwal from IIT Mendi, Dr. Ranjan Dattakolita, Royal Global University, Dr. Yasbir Singh, IIT Ropar, Professor Praloy Maiti, IIT BHU, Professor Niranjan Karak, Tezpur University, Dr. Nirmala Devi from Dona International Institute, Dr. Binoy Sharma from IIT Jammu, Professor Sunandan Borua, Assam Downtown University, Dr. Amit Alexander Naipar, Guwahati, and our Dr. Hare Krishna Dekasar also, because without this resource person, it is impossible to make it a grand success. And at the last, I would like to thank all the Vice Chancellor of University of Science, Technology and Management, Professor Sharma sir, for attending our event, and we are honored that sir has joined as a guest of honor. So at the last, I would like to thank all the participants and all our coordinating team for making this event a success one. Thank you very much.
Thank you very much for everyone.
Now our director sir will declare the meeting as the end with few words.
Thank you, sir, to given us the time to in the valedictory session, and shortly again it is the closing time, but it is a new hope and new vision. Again, we will meet from the 15th of February for the next program. Thank you very much for giving us the time. Thank you very much, sir. Thank you. Now I declare that that program next 5 days, last 5 days is a close. Thank you very much. Now switch off.